or we can do something more sophisticated. This is sort of a notional plot where we have one of these values on the x-axis and another one on the y-axis. And you can see that there are some kind of distribution for y and distribution for x. Each dot represents a variant. And if we were to make a couple of slices here, we could very easily only pull out, say, this peak, if this is really where only the good variants lie. But what if it's more complicated? Well, it turns out that if we have something like a Gaussian mixture model, we can better model what some of the underlying distributions are for the relationships between these two different or many different metadata parameters and values. And we could basically classify them into these larger distributions that resemble Gaussians and basically bin them that way. So it's a little bit more sophisticated than just drawing a straight line. And you end up with these larger categories of the variance that, again, it works a little bit better and is a little bit more refined. But we still need to generate some training data to do this. And that can actually be very complicated, because in order to train something to identify good variants, true variants, you need to have a good true variant data set to begin with. And it's sort of a chicken and egg problem. How do you get good variants when you need to have them to define what your good variants ultimately will be? So historically what's happened is NIST had this Genomes in a Bottle project, which looked at a whole bunch of genomic data and used a consensus calling technique where they would have multiple variant callers run on the same data sets, and they would look at the consensus for all of the variant callers for each variant. And if enough of them agreed, then they would say, yes, okay, this is an acceptable variant, and they put it into the data set. Illumina has a Platinum Genomes project which is very similar, but in addition to doing the kind of processing that NIST did with the consensus calling, they also took into account the family inheritance patterns. So they had the, it was the CEU trio, wasn't it? The trio that we were looking at yesterday in some of our data sets. And based on whether the variants that they found in this family violated the Mendelian expectations, they could then filter these variants based on the expected inheritance values. So that added a little bit more fine granularity. But again, we can even do a little bit better than that. And one of the things that we've started to do at the Broad is to generate this additional data set for true variant calling using a complete hydatidiform, I always have trouble pronouncing that, hydatidiform moles. So for some biological background, what those are is it is a human egg that gets fertilized by a sperm, but the egg has no DNA in it. So it ends up being haploid. And this is actually a medical complication from pregnancy that is actually pretty bad, and it is terrifying. If left alone, it's a medical emergency and it results in death very often. So these hydatidiform moles need to be removed anyway. And since they're haploid, what we've done at the Broad is we've collected a bunch of samples and sequenced them. But we didn't just sequence them with the next generation sequencing method. We actually sequenced them with the long reads methods on the PacBio instruments. And because they're haploid, it's much easier to determine what's real and what's not real, because you never have to worry about heterozygous variants. You only have homozygous regions. And so even though traditionally the long read sequencers are a little bit higher error, it's very easy to figure out what's real and what isn't real, because you should always, for any position, only ever have one true allele. So we do the long read sequencing, we get a whole bunch of contigs, and we assemble them into the complete genome for each of these haploid samples. In this case, what we've done is we've done that twice, one for each of these two different molar samples, and we combine them into a synthetic diploid genome, or a synthetic diploid whole genome sequence. And it's synthetic because it's really from these two haploids, but because it's from those two haploid sequences, we know with certainty what the actual sequence here is. Well, with very, very high probability at least. So how do we use this to generate our data set? Well, we can then mix them up together and run the traditional next generation sequencing on it, do the alignment as we would normally, and then call variants on that data set as we would any other data set that we have. And by comparing the variant calls that we've generated from this short read sequencing to our long read data set that we've created for as the baseline, we can figure out how well our variant calling techniques are doing. And it's a little bit finer grained because we have very, very good knowledge of what the truth actually is. And in genomics, that's always going to be one of the hardest parts. So let's talk a little bit about this Gaussian mixture model method that we have called VQSR, a variant quality score recalibrator. As I said before, it fits a mixture of Gaussians to the features that you've already picked out, those metadata features that I talked about, like depth again and other things. In these graphs, what you're seeing is a two-dimensional projection, but really this is a high-dimensional space, and it's very hard for humans to wrap their brains around that. But as you can see, we've got these Gaussian blobs here that correspond to different categories of variants that we found in a real data set. And in fact, the way this works is these blobs and the surrounding areas for them are the boundaries that we have for the Gaussians. And what we see is the variants that occur both in our data set and the known true variant sites. So it's sort of a way to equate or compare well the variants that we get and the truth data that we've already determined is good variants. So it works reasonably well, and you can, once you've got the regions for the true positives, you can take a look at what's left over, like these, and fit other Gaussians to them so that you can say, okay, the stuff in here is a false positive as well. So we create these Gaussian mixture models that help us categorize these things in high dimensions, and that was the way we were doing our variant quality recalibration, our variant filtering for a long time. But this doesn't capture some of the other details that we might have in our data sets. For example, we've already told it what the features should be, but some of these features aren't modeled very well by Gaussians. The mapping quality, for example, we use a hard cutoff at 60. Above 60, we don't report any higher numbers, we just leave it at 60. So you end up with this, if you were trying to fit a Gaussian to it, it wouldn't work because you'd get to the peak and then there's just a drop off at 60. There's this hard cliff. It's sort of a toy example, but it helps to illustrate this a little bit more. But there's also the fact that we've picked out these metadata parameters already, but what if there's more to the story? What if there's some kind of phenomenon that we haven't captured in a value that would be important for calling variants? And as an example of that, we could talk about sequence context. Now, this is a Lego plot. I don't know if, has anyone seen one of these before? All right, so the colors represent the variant changes. So yellow is when a C gets turned into a T. Purple is when an A gets turned into a T, and so forth and so on. In each of these different quadrants, the context around that SNP is represented by these. So for example, This green corner segment is when an A got turned into a G, and it was G, and then that original A which was turned into a G, and then followed by a G. So the sequence before the variant, the reference would be GAG, and the sequence after the variant would be GGG. And you might think that the context shouldn't matter, that this should all be flat and everything should be equally probable. Well, that's not actually true, and it isn't true for a few different reasons. So if we look at this really high purple peak here, where it's A to T and the sequence is TTT, or T around it, well, that's a homopolymer repeat, and traditionally our sequencers don't do a great job all the time at homopolymer repeats, so maybe that's an artifact. But maybe there's something else going on too. It's not always clear that this would represent an artifact or that it would represent some underlying biological phenomenon. There, a priori, isn't actually a good way to tell, but we know that these are the distributions, and we know that this has a whole lot more, this particular context has many more SNPs than we would expect. We don't necessarily know why, though. So what do we want to do? How do we put these data into our model, and how do we model it? Well, we can encode the DNA in the reference sequence to start with. What we can do that with is with a tensor that has this one-hot encoding. And what that means is if we have a sequence of DNA up top, this is a Tata box, the way we can encode that is in this tensor with 4 different channels, 1 for each of the bases. And it's one hot because in the A channel, if the base in the sequence is an A, you get a 1, and if it's not, it's 0. And so we can encode this sequence into this one-hot encoding, and we can also include some ambiguity there with this N base representing any of the 4 bases, which you would expect to be about equal for each of them, so 25% since there's 4, and this K representing the G or T channel, which means that there's a 50-50 chance that it's a G or a T, but we know that it's not an A or a C. And these actually are, it's a little bit into the weeds, but they're IUPAC base representations, which you might find out about if you're looking at the reference sequence or some other sequencing techniques that don't necessarily know all the time what the exact bases are. But these are other ways of representing the possible bases without knowing the exact bases. And so what do we do with that? Well, traditionally one of the ways that we would find these motifs and figure out what the different little sequences are is we would do a pileup, a multi-sequence alignment, so we know they all start and end at the same position, and we can just easily check what the most frequent base is, and we call that into a consensus. So it was A, C, G, T. So we have the A channel, C channel, G, and the T channel. And we have the consensus represented down here with the bigger letters being the consensus for that position. So as you can see at the beginning, it's a, oh, I forgot myself. It's a T, and then, and so forth and so on. But just knowing what the most likely base is doesn't tell us the whole story either. It might be nice to know what the probability of those positions were. So we can then go from this overall consensus into this position weight matrix that scores what the, how likely each base is at each position. And well, where does that bring us? What we can do then is we could take our sequences and we can use convolutions to basically find where these motifs are. Yes?
Minus infinity is that there's no chance of that in happening, like a null.
Yes.
Oh, okay.
Yeah, so you'll notice, what is the 1, 2, 3, 4? In the 4th position, there aren't any A's in that sequence at all. So for A there, yeah, it would be minus infinity. So now that we have our sort of method of storing the data in memory and working with it as and letting the algorithm work with our data, what we can pull out are these motifs in the sequence. And the way we do that is by convolving these one-dimensional sequences over the genome. And what that basically acts as is a motif finder, whereas the motif is closer and closer to being an exact match, you get a better and better score effectively. And what we're actually doing is we're allowing the machine to learn what the motifs are as part of this process. Now, it'll take a little bit of explanation, but this is basically what one of the layers of our neural net is learning, one of the early layers. And what this basically says is, again, the bigger the letter is, the higher weight it has at that position. This is a poly-C detector. So it actually is able to pick out that there are these features that we've associated with higher than expected variant calls, like homopolymer repeats. And so looking at the reads and a whole bunch of data about the reads encoded gives even more power to finding information about variants and being able to label them, but it's also much more expensive, as you would expect. So here's an example of what the input tensors look like at this point. It's not just the reference information, but it's also the read information, whether it's a deletion, whether it's now, to store all of this information in different layers. But now that we're storing all of this info, the machine learning algorithm, our convolutional neural net, has a lot more power based on the raw read data to identify whether our variants are good variants or whether they're false positives or something else. So here we can look at, in a couple of slides, some of the details about what these look like in the tensor space. And it's a little bit hard to tell, but what we've centered around here is G2&A SNP, and we've got all of the read context and reference context around it as well. So what you'll see is right in the middle, this orange transition, and I almost said transitioning, that's not quite right, this orange, and then this green, representing the G and the A. There are a bunch of these, but I find these kind of hard to look at and hard to really appreciate, but ultimately this is what the neural network is looking at and how it's encoding these things when it actually does the evaluations. So for a couple of other examples that are very similar, we've got some insertions and deletions that are represented by, when there's missing data, it's represented by this purple. And so we've got this deletion 1/2 of the genome of this region here, but it's still present on the other strand. Similarly, we've got a homozygous deletion of TTA here, and that's represented by this purple. And the insertions are very similar, but the purple is in a different place because it's an insertion rather than a deletion. This is all to say that the read tensor can encode both SNPs and indels, and we learn them at the same time. With VQSR in the past, what we've had to do is come up with a model for SNPs and then a model for indels, because the data types are different enough that we wanted to be able to properly model what a good SNP or what a good indel looked like. With the neural net techniques, we learn them all together. And that makes training take a little bit less time, although in practice, we don't have to worry about training at all. And so what we now have is this CNN variant or CNN score variance tool. And this is what the neural network looks like. We have all of this data that we put in at the front, and there are several convolutional layers. Then we add in some of the read annotations, we merge them together, and then we've got a multi-layer perceptron here, which is just a fully connected neural network at the last few layers. what we get is ultimately a single value that we can then filter on, but it's this sort of composite that you could think of as how confident the CNN tool is that it's a real variant or an artifact. And so how well does this perform? Well, the way we can look at this is if we look at the area under the precision recall curve, it should be 1 if it's perfect. And it's pretty good. I mean, we do pretty well. I hate to call out competitors. Deep Variant does reasonably well too, but we're doing pretty well at identifying these things and scoring our variants properly. Indels being the harder case, it's difficult, but yeah, this technique works.
CHM Indel set has long events that would call up the long read data that we can't get with short reads anyway. So there's sort of a maximum sensitivity even for perfect calling short reads.
Yeah, that's a fair point.
There is a different reads in between the samples because in the Indels, because there is one sample CHM where is the data of that reads. You have minor probability to CNN with GATK code. I don't understand that difference between the.
Why this is lower than this? Oh, that's what Laura was just saying. So in the CHM synthetic diploid data set, because we sequenced it with long reads, we're able to tell where much larger insertions and deletions are. And because the short reads have a much shorter, like they're 150 bases long or so, some of these insertions and deletions are much larger than that. So there's no way to know with certainty that that's actually a real insertion or deletion. So we're missing some of the really, really big stuff.
There was also the early versions of that data set didn't have one base indels because of the noisy long reads, and those are the vast majority of the events and also the easiest events. So it's kind of a handicap if we don't include those.
And another way that we can validate this is by having a whole bunch of data sets that we train on individually and then see based on training on one data set, if that training is applicable to other data sets. So this is a matrix of one data set on the x-axis and one data set on the y-axis. So data set A is this CHM HG38 SynDip, whereas G is NA12878 on HG19. And so with this CNN tool, we can train on one data set and then see if that training will generalize to other data sets. So if we're really, it's a good way to figure out if we're really learning something about the nature of variants in the human genome. And we can do this across HG19 and HG38 and different sequencing platforms and different samples. And column K is sort of the generalized learning across all of them. And while learning on one might not be directly applicable to some of the others, if we learn on all of them, then we're able to identify very well what good variants are and what bad variants are. Here, as you'll see across the diagonal, 0 is sort of the ideal. So farther from 0 is worse, and that's why these are labeled in red. And we're pretty close to 0 in most cases here. So it does seem to work, and it does seem to generalize across different data sets, which is really nice. So again, we have this 1D CNN and we have a 2D CNN. The 1D model uses our annotations like VQSR did, but it also uses the reference sequence. The 2D model uses those two things and the read information as well. And this is the way that you would call either of them. The 2D model takes a little bit longer, but still works well. We've in fact trained these models already, so if you run our tool, you could take advantage of the training that we've already done. And again, I should say, this will annotate each of the variants with that, again, I'll call it a confidence score of how likely it thinks it is to be a true variant versus an artifact. And then we can use this filtering tool to filter out based on that single aggregate score afterwards. So yes, if you look at it at a high level, it's hard filtering, but it's hard filtering based on this composite value that has a lot more power behind it. And it's not just hard filtering on these single attributes, it's hard filtering on this really well-learned composite criterion.
I would argue it's actually not hard filtering because it uses the truth sites from 1,000 genomes to set the threshold. So you don't set the threshold yourself. You don't make that decision. You let the software set that threshold based on data that you do believe and what you want your target sensitivity.
Okay, yeah. That's a fair point. Some of the responses people have had are, well, you're filtering again, but you're right. It's more complicated than hard filtering on its own. I think that's it. So we've gone through filtering our variants, and now we can talk about refining genotypes and then annotations after that, and doing some call set evaluations. Does anyone have any questions?
Which libraries are you using to train and build models?
PyTorch, I think. The CNN tools are largely Python-based. At one point it was PyTorch. Laura, do you remember what they're using? PyMC3 now?
It's a little bit older.
Okay. Yeah, it's sort of a, yeah. It's sort of a dirty secret. The GATK is written in Python, but a lot of the machine learning stuff we're using is GATK is written in Java, I'm sorry. A lot of the machine learning stuff that we're doing is obviously in Python because that's what the machine learning community uses. So we have a bunch of Python code now in GATK as well that is doing all of this. And again, if you download the source code, you can take a look at it. Okay.
All right, so the last thing that we're actually going to do to our variants is the genotype refinement, and we did a little bit of that yesterday in the hands-on tutorial, although kind of out of sequence, but that's okay. So the real sequence does involve the filtering that John just talked about before we do the genotype refinement, because we don't want to waste effort changing the genotypes for sites that we don't believe in anyway. So a lot of people care about variants, but does it really matter what the genotype is? 1 copy, 2 copies, like, is that as important? Medical genetics definitely needs to know how many copies they have. If there's a loss of function variant, do you have 1 functional copy? Do you have 0 functional copies? And it's also important to know what the parents' genotypes are. We've seen a lot of cases where parents have a child with some sort of developmental delay or some sort of structural malformation, and they want to know, like, if we have more children, what are the odds that this is going to happen again? If it's a de novo mutation, the odds are really, really low, and so it'll probably be okay. If it's a recessive mutation that both parents have, then they're probably going to want to see a genetic counselor, potentially do some pre-implantation genetic testing. And we also use them in the larger studies, the big common variant population GWAS type studies that Kate's going to talk about later, because we want to know how many copies each person has in terms of the dosage. So if a person exhibits a phenotype that's maybe increased height and they have 1 copy of the allele, do we expect people that have 2 copies of the allele to be even taller? But those are the important questions that we want to know. So we looked at IGV yesterday, and it'll give us information about the variant call at the site level and also at the sample level for the genotype. For example, there we're giving the alternate base T, as we see in the reads as well. We have the annotations that we use for filtering as well in the site level data for that variant. And then if we look at the sample track for the VCF, then we get all of our per sample information with the genotype. It tells us verbally what that is, heterozygous, homozygous variant, and also the genotype quality, attributes, the allele depth, and so on and so forth. So that's a pretty good looking variant, right? I think those might be our artificial haplotypes, yes. So of the actual reads, like these are the assembled haplotypes like we looked at yesterday, but in the actual reads, we only see Ts. It's a good looking variant. Genotype quality 63, less than 1 in a million chance that we're wrong. Looks good. But not everything looks that good. So if you have lower coverage, if you have some observations with poor base quality, if you're looking at indels in repetitive regions, all of those can be harder to call, require more evidence, and generally have lower confidences. But we can use additional data to improve our genotype calls. We can almost always use population data, like 1,000 genomes. We've shown that having population-specific data, for example, we looked at variant calls on a Finnish population with population allele frequencies from a larger Finnish study, does great. Using the 1,000 genomes with the aggregation across all populations, African, Asian, Central American, European, still does a pretty good job. So even if you don't have Costa Rican specific allele frequencies yet, then you can still use 1,000 genomes or some other population to help refine your genotypes. If you're doing a family study, particularly a trio-based study, then you can use that information from the parents to inform the genotype of the offspring. And in some cases, depending on the genotype configuration, you might be able to inform the parents' genotype likelihood as well. And so we can apply those additional sets of information as priors to modify our genotype likelihoods to produce better calibrated posterior probabilities on each of the genotypes. So this is my metaphor that I love, and I actually saw that it was true on Monday. So imagine that you're in the lab, and it's not like this beautiful new building where you have awesome windows, you're in the basement, and you can't see outside. You don't know what the weather is. But your coworker just walks in carrying an umbrella. And so you say to yourself, well, I mean, usually people have umbrellas when it rains, right? So having observed that your coworker has an umbrella, what's the probability that it's raining outside? And if you're in Boston and it's the spring or the fall or sometimes even the winter, then the probability that it's raining, the prior probability based on what we know from weather in the past is reasonably high. If you're in Costa Rica in the summer and it's the dry season, then the probability that it's raining is actually pretty low, right? Well, it's lower than other times of the year.
It could rain iguanas in here.
Could rain iguanas. You need a much bigger umbrella. But maybe in that case, so the posterior probability, if you have this very, very low prior of observing rain at this time of the year, is still probably pretty low. And maybe it turns out that your coworker is carrying an umbrella because she doesn't want to get sunburned. So here's our full workflow for the genotype refinement. We start with the recalibrated variants, which come out of VQSR or CNN. We can apply population priors or potentially family priors if we have related samples and a pedigree to go with them. We run this tool, calculate genotype posteriors. That's going to yield new genotype calls potentially according to the posterior probabilities of those genotypes. And then we recommend that you run those samples through variant filtration, which will filter out the low quality genotypes because you don't want to use them for a lot of analyses anyway. And then we can run variant annotator, which will call out specifically variants that are de novo if you have family data. Calculate genotype posteriors command looks like this. It takes a reference and optional pedigree and then a VCF with the allele frequencies population you're using. If you have a large cohort, it'll actually also use the genotype calls within your cohort to help calculate genotype posteriors, sort of an expectation maximization algorithm. So here's an example from our beloved CEU trio with 1,000 genomes data. So initially, the call for our beloved NA12878 was homozygous variant. And then we applied priors from 1,000 genomes, which have a particularly low allele frequency at this position. And then we see that the posterior genotype call is heterozygous. So you can see that from the allele frequency and also from like the dark blue for the genotype call itself. And if we look at the BAM at that particular location, we actually see that, yeah, we've got a pretty high proportion of reference reads. It's not a beautiful 50-50 allele balance, but it's very unlikely that it's homozygous variant. And so in that case, the original likelihoods that we started out with weren't particularly confident. So we're looking at like GQ3, not great. But then if we apply the priors based on the 1,000 genomes data, we don't see this allele very often in any other sample. So the posterior probabilities are now that it's a different call, heterozygous, with a higher confidence. So we went from GQ3 to GQ27. So that's a variant that we believe in, a genotype that we believe in that we can use for a lot of different analyses. It works the same way in the other direction. For example, the initial het call here had likelihoods that yielded just GQ0. It's a coin flip, whether it's het or hom var, we don't know. So if we apply population allele frequencies from 1,000 genomes, apparently this is an incredibly common variant, a suspiciously common variant, such that maybe the reference is wrong. But if we apply those priors, then we see the posterior probabilities show that it's most likely a homozygous variant call with improved confidence at Q16. So still not a great call, but it is in agreement with what we see in the reads and what we see in the NIST genomes in a bottle database. And most importantly, sometimes it'll change the genotypes like we saw in those two examples, but just being able to get a more accurate quality score is still very valuable for our analyses because we see that the calls coming out of haplotype caller aren't particularly well calibrated. And so if we take all of the homozygous reference calls that we assigned a genotype quality 20, for example, and then we compare those to some truth data set, we can see what the actual error rate is. So if they have genotype quality 20, GQ20, you do the Fred scale, yada, 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 that's a 1% error rate. So you expect to see one error in 100 over all of your genotype quality 20 calls. If you evaluate those against some truth data source, we actually see that the homozygous reference calls in that top panel are underconfident. So those are, initially we're showing those in red. So the empirical measured genotype quality is higher than the reported quality. So there's actually a lot of data there that we could be using, but it's not being correctly assigned a quality that we want to use. So once we run our genotype, calculate genotype posteriors and recalibrate our genotype qualities, you'll see that the posterior calls lie very nicely along that x equals y line, showing that the measured quality is in very good agreement with what the reported GQ is. Heterozygous calls, they do pretty well out of haplotype caller. We don't gain a lot there. But then for homozygous variant, we're typically overconfident. So in the initial red calls, we see that the reported quality is higher than the empirical quality, which you can tell because it's underneath that x equals y line. But then after we run calculate genotype posteriors, now they're lying nicely along that x equals y line, meaning they're in good agreement with what we see in our measured data. So on the whole, this clearly benefits a lot of cases, but what happens for incorrect calls? Do the genotypes get better? Do they get worse? Do we become more confident in things that are wrong? So if we stratify our calls by things that are in agreement with our truth data or not in agreement with our truth data, for the most part, the correct calls get a Q10 boost in their genotype quality, which is great. So if your threshold for using a genotype in your analysis is GQ30, then now things that are GQ20 and would have been filtered out initially are good enough that you can actually use them, so you get more data. Things that had an incorrectly called genotype actually stay about the same. We may not have information from the population to inform those, but we're certainly not making them any worse, which is great. So how do we actually do these calculations? We can use the patterns of Mendelian inheritance. So we know that the child can only inherit alleles that are present in the parents. Simple enough. So there are several valid combinations of genotypes and invalid combinations of genotypes for the whole trio. For example, if the child is homozygous reference, then both parents could have been homozygous reference, one of them could have been het, the other one could have been het, or they could have both been heterozygous, in which case there'd be a 25% chance that the child would be homozygous reference, but it's still possible. And we actually do use those probabilities in the calculation. If the child is heterozygous, unfortunately, there are a lot of different combinations where that's valid, and so we don't have a lot of power to correct heterozygous genotypes. But if the child is homozygous variant, it mirrors the reference case. So haplotype caller gives us the genotype likelihoods, which is the probability of observing the child's genotype given the data that supports that genotype from the reads. But if we have data from the whole trio, then we can get the probability of the child's genotype given data from the child and the mother and the father. So this is in accordance with Bayes' rule, which is sort of the premise for a lot of the more sophisticated math that we have in the GATK. So what we're going to do is we're going to look at the likelihoods, which is the probability of the observation given whatever the unknown state it actually was, and the prior of that state. So where we're going to use the priors on our genotypes from population information, family information. And we normalize that to get the posterior probability of the data, the genotype, given the observations, the reads, and also the population information. So we can apply priors based on family information by using these Mendelian violation calculations. So we use mu as the rate of de novos, effectively, which admittedly varies depending on what you see in the literature, about 1 in 10 million. And so if there's one Mendelian violation, we say that we'll give that a penalty of that same de novo probability. If there's two, then there was one de novo and another de novo, so we'll multiply that probability against itself, squaring it, and then just subtracting all those possibilities from one. If it's not a Mendelian violation, then we have this other probability that's based on how many configurations we have with one violation and two violations. So then we're going to go ahead and apply these priors, where again, these are our genotype likelihoods, and then we have our prior based on the likelihood of the father, likelihood of the mother, and the prior on the genotype from other information, and we normalize that to get the overall posterior for the child. So like I said, we expect from the literature that the mutation rate is about 10 to the minus 8. But in reality, that's a little bit strict. So tuning that parameter, if we start with the original 10 to the minus 8, it's a little bit too strict. So our true positive rate for the CEU trio data is pretty low. It's actually only like 40%. We're penalizing a lot of sites. So in practice, a better value for that parameter is closer to 10 to the minus 6. And so you see as we increase the probability of seeing a de novo mutation, we can increase our sensitivity because there seem to be more de novo calls in the data than we would expect. The next step is variant filtration, and we're actually going to be filtering at the sample level rather than at the site level based on the genotype for each sample. And so based on what other people at the Broad have done for their family analyses, we filter out anything that's less than GQ20. We give it a filter name low GQ because that's 99% confidence. If there's more than 1% error rate, like, do you really want to use that data? Maybe not. You could go even more strict with maybe you want to filter out things less than GQ30 if you're doing, if you're going to do some time-consuming follow-up on the data that you do get, so you can kind of restrict your results to just the best. From there, we run variant annotator, and the annotation that we're going to run, I called possible de novo, where we're still not 100% sure, but like we're going to flag it so you can look at it. Do some more investigation later. So specifically talking about de novo mutations, they're obviously really important in a lot of Mendelian genetic disorders, rare disease cases, and we expect to see about 30 de novo mutations per genome. And again, this is the scenario where both of the parents have reference copies of the allele and the child somehow acquires one copy of an alternate allele. Maybe a mutation in the germline, maybe a mutation as the embryo is developing. It's hard to pinpoint where, but at some point, something went wrong biologically and now they have this new allele. So what do we want to be able to say about the de novos that we find? We want to make sure that the de novos that we're confident in are novel. So the child should be the only alt allele in the trio. And a lot of people say that across the whole cohort that they're analyzing, they want that to be the only alternate allele. So some analyses are a little bit stricter, but we do want that to be rare in general. If it's truly a de novo mutation, only a de novo mutation that's disease causing, we shouldn't see it in that many other samples because our other samples are going to be normal, developmentally typical, and so on and so forth. And we want to make sure that there's a high genotype quality for both the parents and the child, because the most common error mode is that we miss the het call in the parents. So they did actually have an alternate allele, but as we sample the reads off of those two haplotypes, every now and then we'll just get unlucky and we'll continue to sample from the reference haplotype without seeing any alt reads. It happens. But it's less likely to happen if we have more reads in the parents with a higher genotype quality. So based on this real data from an analysis I did a long time ago, they had a rare disease scenario, a suspected Mendelian disease, and from the initial calls in the trio of exomes, there were 417 de novos from the recalibrated, variant filtered calls before we used any of the family information or population information. Once we applied the family information, population information, we got that down to 17. Again, these are exomes, and so we don't expect to see very many at all, like maybe 2 in unhealthy samples. If we filter then by genotype quality to just the highest confidence, we still have 8, which is higher than we would expect, but still a small enough number that you can go and do your functional annotation and narrow them down by the consequence, and then look them up in the literature, see if that gene that has the mutation is associated with any of those kind of phenotypes. 8 is a very tractable number of variants to work with, whereas 417 is a lot of analysis work. So we've gone a long way towards saving manual review effort for analysis. So
After genotype refinement, we expect to see more high-quality genotypes, because the initial genotype calls could be low quality, totally ambiguous, or entirely wrong. But we can apply population information in pretty much all cases and family information in some experimental designs. And if we have more high-confident genotypes, then that's more data to look at, more variants to run associations on, more potential disease-causing variants to discover. And like I said, if you run functional annotation on those, then you could potentially filter even further if you're only interested in things that are loss of function, for example, or things that are missense, because it's incredibly rare that synonymous mutations actually cause disease, although there's some cases like progeria, which is pretty darn cool. Biology is weird. So at this point, we have our filtered variants. We've improved their genotypes using population information, maybe pedigree information. And then we have done our functional annotation, and we're ready to evaluate our call set. So did we do a good job or not? And John will tell you how to do that. Are there any questions about genotype refinement?
Check, check. Good? Okay. So now we've got our data sets, and we want to see how well we did at calling variants and whether we can do good analysis on it and our data are good to be studied further or published. And this is pretty much the last step before you either troubleshoot and reevaluate and go back to some step earlier in the process, or use this data to do some meaningful scientific study. And it's sort of a spectrum. If you find a whole bunch of variants and you match them perfectly to some underlying biological truth or phenomenon that you're expecting to see, then you have an ideal case. And maybe sometimes you have some false positives, and then if you only have artifacts, it's pretty much the worst it can be. Often, or almost always, we're somewhere in the middle. We'd like to be further on this end, but that's just, biology is hard, and genetics is a hard problem to solve. How do you know whether you have a good call set or not? Well, you need to have a truth data set to compare to the data that you have. And it's similar to being able to score the variance, but in this case, you want to be able to actually do a direct comparison with a data set that's very similar to your own. And you can look at different measurements across these two different call sets, the truth and your call set, to see if you've done a good job. And if the measurements are very similar, then you probably have. But like I said, the data sets need to be similar. In particular, the population ethnicity will widely change what certain values for these criteria are. If you do a comparison between the number of SNPs expected versus in a European sample versus an African sample, you're going to get two different numbers. And that might be alarming, but if you were to compare a European sample to a European sample, it would be much lower. Similarly, if you were to compare an African sample to an African sample, it would be much lower because you're comparing a similar data set. Along those same lines, you want to compare genome to genome samples and exome to exome samples, and the cohort size, the total number of samples in each group should be roughly the same as well, so you have a good basis for comparison. Sometimes you might not be able to do this easily, and you might have to split up some of your data to do sub-comparisons between a small portion of your data set or a small portion of a truth data set that you're able to match up in a way that's easily comparable. And to sort of illustrate the point for ethnicity, these are the number of singleton SNPs per genome for a whole bunch of different ethnicity backgrounds. And you could see that it varies quite a bit. So we have a bunch of truth sets that have known variants in them. DBSNP is kind of this huge database of pretty much every SNP that's been discovered goes in there. So there's a lot of stuff. You may not want to use it. EXAC for exomes and NOMAD for genomes is pretty similar, sort of. They have a lot of variants in there from many, many different studies from human data. And there are other more highly curated data sets that you can use as well to do the comparison with. I talked a little bit about genomes in a bottle and platinum genomes earlier. These are examples of things that you can use as truth sets for your call set evaluation. And again, to drive home the ethnicity point, these are a bunch of different studies separated by these dotted lines. And the different colors represent different populations where each dot is a variant. And it's just a count, or each dot is a sample, I'm sorry. We're just plotting a count of the number of SNPs that were found in these exomes. And you could see that it varies very wildly depending on what the background is. What you may notice is that the European samples have, by and large, the fewest number, and that's because the reference is largely based on European genomes. So the number of SNPs will be higher for populations that are not European. And you can sort of ascribe this to a founder effect where older populations have more genetic variation because they've been settled in the same place for longer. And then if you have a group that migrates out to someplace else, like an island or to Europe or wherever, they bring a very small subset of that genetic diversity with them. And then that will branch off eventually into other kinds of diversity, but the other diverse samples will remain in place because they didn't do the migration. So this founder effect will propagate subsets of that genetic diversity down the line. So we have a few different metrics that we typically recommend for doing call set evaluation, and I'll walk through them pretty briefly, but they're relatively straightforward. The first is the number of SNPs and indels that you have in your data set. And it's sort of a sanity check. It's a high-level check that you would have. For a genome sample, as you can see, you would expect about 4.4 million, and an exome about 21,000 variants in a single sample. And this is a quick litmus test. If you diverge greatly from these numbers, then there might be something you need to look into in terms of what you were actually looking at, or doing more refinement on what your variants actually are. Another one is the indel ratio, so the ratio of insertions to deletions. And you would sort of expect a deletion and an insertion to be about equally likely, so the ratio should be about 1:1, or overall 1. And in fact, we do see that for common indels. But for rare indels, what we see is that deletions are much more common. And that seems to be because of the selective pressure for novel deletions hasn't had enough time to normalize yet. But these are sort of the expected values for common variant, common indel, and for a rare one. And again, if you see a divergence between your values and these, then it warrants further investigation. Transition-transversion ratio is also interesting. And this is human-specific, but a transition is when a similar base turns into its mate. Not a mate in terms of what they pair with in the genome, but a mate in terms of the way the molecule looks. If you go back to your molecular biology, A and G are very similar, and C and T are very similar. And so a C and a T are actually much more likely to trade places, and an A and a G are much more likely to trade places. That's what we call a transition. A transversion would be if an A moved to a C or a T, or if a C moved to a G or an A, and vice versa. The rate at which that occurs is a transition is about 4 to 6 times more likely than a transversion. And the ratios end up being, for a genome, about 2-ish and then about 3-ish for an exome. So if this number is off, then for SNPs, obviously, because it's just a single change, then you might have a whole lot of false positives if the number is too low. Then the last one is genotype concordance. And Laura was just talking about refining the genotypes, and it's it's a pretty straightforward comparison. You just check to see if your calls have the same genotype as the truth for a particular variant. And it's measured as percentage of which ones you have and which ones you don't have in your data set. If you don't have something that matches, then we would say it's a false positive. And that would count against you for an overall score. Now, the best way to get the truth data from which to do your comparison is by some orthogonal measurement method. So you're not compounding your artifacts from sequencing or from the way that you're creating your data. And you can do it by, if you're using an NGS sequencing platform, you can use a different platform like Sanger sequencing, or even just a different sequencer altogether, or long reads or something like that. You could also use targeted sequencing if you want to jump right into specific loci and use those as your reference points. One of the things that can help is even adding data from known samples into your data set when you are sequencing, so that you can have some additional information going through the same method that you're using, and then compare it back. So to quickly jump into a kind of toy example, we've got a nice new call set that we have run through our system, and we want to take a look at our variants. And then we have this gold standard variant data set that we know these are the good variants, and we want to make sure that we have all of these in our data set. And this is how we're going to actually do our comparison, whether the call set is good. So we can measure our sensitivity, which is just the true positives over the true positives plus the false negatives. So the false negatives being the ones out here, and the true positives being all this data in here that we found in both the gold standard and our call set. For this, it's about 70%. Our false discovery rate, which is the false positives over the false positives and the true positives, is 42%. So it's 42%. It seems large. And then we can look at genotype concordance, where we take a look at whether the genotypes match up in the gold standard at the variant sites to what we have in the call set. And these ones in the boxes match up correctly. So we've got 4 matches over the total number of true positives, 57%. And that's kind of a high-level overview of how we would calculate some of these numbers. Running these things with GATK is straightforward, and this is how you do it. For variant evaluation, you can call our variant eval tool with the reference, and you have the call set you want to evaluate, the truth data set you're comparing it to, and then you give it an output file. Similarly, you can call genotype concordance to do the genotype evaluation. And again, it's similar, but the data, the names of the inputs are different, where your call set is now call VCF and the truth set is now truth VCF. And you end up with another output file that you can take a look at to evaluate. Now, once you have evaluated your call set and you were able to take a look at the statistics, often this is when a whole bunch of people will get in a room together and argue about whether it's good enough to actually do a study, or whether you have to go back and redo something because the data aren't quite good enough yet. But at this point, we've gotten more or less to the end of the pipeline where you'll either figure out what went wrong or go on and do more analysis to find out your medical anomaly of interest. Any questions about evaluation?
It can also be used for panel of genes, not only West or WES.
Sorry?
It can be used in panel genes, like 15 genes or 25 genes.
Yeah. So you could do a panel as well and do a similar analysis with that.
Vary the threshold of the
right. Well, with that, I think we have a short break.
Yes, so we're gonna have a coffee break until 11:05. I believe we're upstairs again for that?
Yep.
Fourth floor. So we'll be back, with some
brand new terms of service. We'll go back to, not workspaces. From the,
Actually, they should already have it cloned, so you can just go to workspaces.
oh,
You should be able to see it.
good, okay. Yours is here, great, cool. So yesterday we did germline variant discovery, where we did haplotype caller, and then skipped ahead and did a little genotype refinement, and now we're gonna talk about filtering. And there's two different filtering methods that we're gonna look at today. The three big ones are VQSR, hard filtering, and CNN, where VQSR is something we run for at least, probably at least 50 samples, maybe, 20, 50. But hard filtering and CNN are methods that also you can use on single sample data, which is why we're gonna experiment with those today. So we're gonna start before lunch with hard filtering. And your runtime is going to use the same settings as we did yesterday. So that should be good to go.
When you click on your notebook, it's gonna pop up a screen, and instead of saying start your notebook, it will say edit or playground. You'll wanna click edit, and that's gonna start, unpausing your runtime, so just click edit. While we're waiting for it to start up, the two different modes you have to work in, you have edit, you have your edit or you have your playground mode. The edit allows you to change things that happen in there, whereas playground mode, when you make changes to it, it won't save. So if you're planning on editing something, you want to use edit mode. If you're working in something, you just want to like test it out, you can use playground mode. For our purposes, you could use playground mode, but I figure you might want to save what work you're doing in it for later.
And there's differences when you're collaborating on the same notebook, right?
Yep, so if you have multiple people in the same workspace, which you can share your workspace, only one person can have it open in edit mode at one time, and that's to prevent you from overwriting each other. So you might use playground mode to test things out and then go over to make the changes you want to make in the main copy.
So as part of this tutorial, we are going to look at all those filtering annotations that we use for CNN, VQSR. We'll talk a little bit what they mean, and we'll plot them so that we can find good thresholds for each of those, several of the informative annotations based on truth data from the genomes in a bottle. So we're effectively going to be cheating a little bit today because we're already going to know what the good variants are, and we're just going to try and set our filters so that we can capture the most of those known true positives. For a real data set, you won't have that advantage, but that's kind of why one of our recommendations for call set evaluation is to include some truth samples. So if you're doing your joint genotyping and you want to throw in a VCF from NA12878 that you get from us or from somewhere else, then you can still use this method. If you even do sequencing of NA12878, then you can still do that. But it's really helpful because otherwise you're effectively just flying blind based on what looks like a pretty distribution, but you won't know actually what the true positives are and how good a job you're doing. So the runtime should already be set up. We're going to use a bunch of CPUs so it's a little bit faster and a little more memory. And in this notebook, we're going to be running mostly in Python, but the plotting is a little bit easier in R. We particularly like this library called ggplot. So we are going to use this additional library to run R from inside Python. So I'm going to shift enter those. The second one took a little longer than I expected, but hopefully yours was successful with no problem. And then we're still going to be using Python 3, so we just want to make sure that that is still set properly. So we're going to go to kernel, and what do we do? It should be up in this corner, yeah. So we used Python 3 yesterday, should still be Python 3. Cool kids don't use Python 2 anymore. So.
No more shit.
And we're going to go ahead and define our workspace variables again like we did yesterday. So set a variable for our crazy bucket name and for this workshop iteration so that we can pull the files specific to today. And then before we download all of the files we're going to use, we're going to make directories on the notebook file system. And same as yesterday, we'll check to make sure we can actually copy those files. Should be perfectly fine. Taking a little longer than I expected, but I have my list of files. Looks great. Everybody see all the tutorial files in the server with those 7 or 8 paths? Okay. And now we'll actually localize those files to our notebook environment. All right, so while my file's download and your file's download, we'll talk a little bit more about what we're going to do. So we're just going to work with SNPs for today. All of these same strategies apply to indels. The data is just not as clean because there's reference bias, because there's repetitive sequencing, because there's PCR error. The indels just are not as well behaved, but all of the annotations that we have for them are still informative. It's just the distributions are not going to be as clean. So for the purposes of showing you how to do this in the best case scenario, we're just going to look at SNPs. So does everybody have their files downloaded now? No. Are we still waiting or are we lost? Oh, I think Kate is on the fancy schmancy network, not the guest network. But it's spinning. It's not spinning. Okay. Right, so this is the top of our VCF, and you'll see we only have the one column for the one sample that we were interested in, and all of the ACs, the allele counts and the allele numbers make sense as we would see for one sample. I think the rest of the annotations are the same though, for the full cohort. Right, and it's got her genotypes, and all of her genotypes should be hets and home vars because we pulled out the home refs, and of course our broken pipe warnings that aren't really important. So we have our SNPs, and then we want to label them with their truth status, and we're going to use a VCF from the NIST Genomes in a Bottle. So the resource line is going to give the file name and also the name that we want to call the thing, so what we're going to annotate the actual line in the VCF with, what we're going to label it with. And we're going to give that label whatever's in the call set's value of this resource. So this -E expression is with relation to this resource GIAB. And hopefully that'll make a little more sense when we actually look at the output. It's actually not in beta anymore in the latest version. We took care of that. So next workshop.
So 4.1.4.2.
Yeah. Yeah, and concordance in the other one we got. I made them take out of beta too. All right, so that's done. And we'll take a little peek at the top. And so here is the new data that came from that variant annotation command. That's our giab.callsets. And they're all integer numbers because the Genomes in a Bottle resource is a consensus across multiple call sets. GATK was used, possibly 2 different GATK variant callers, like an older one. I think they ran SAM tools and maybe Freebase. And the theory was that if enough call sets agreed that that variant was a true positive, then it's probably a true positive. In all fairness, in that way, this data set is a little biased towards easier events because tools that did a good job calling the harder events and thusly were better than the other tools won't have any other support from the other resources, which is one reason we prefer to use the synthetic diploid CHM data for a lot of our analyses. But for our purposes here, these are very, very high confidence variants because all the different tools called them. And so exactly how many different tools called them is given by this call sets number. So to get ready to do our plotting, we're going to run this variance to table command, which is going to parse the VCF and put it into a format that's going to be easier to read in for R. And all these -F fields are giving us just different fields in the VCF. So if we look at our VCF header, yeah, so we're going to take the chromosome, position, the qual, and then these refer to specific keys in the info field, like this base quality rank sum. We're going to use depth and the Fisher exact test on strand bias. And then, of course, we're also going to output this call sets number that we just used to annotate. And then we're also going to use the genotype quality. And so that one, let me scroll up a little bit. That one has a slightly different argument because it's a genotype field. So we only have one sample. It's just going to output it for our one sample. But if we still had the husband and the son, then we would get a GQ value for each of them because that's per sample information. So I stare at VCFs all day. This other format doesn't really bother me. I can read all those, but it's a little bit easier to read if you put it into tab separated with column headers, and that's what we're going to use to read in for plotting. So this all looks good. We don't have any NA values. If you had a typo, for example, and you put like lowercase dp and it didn't find it, then the whole column would be NAs, and you'd say, oops, something's going wrong here. All right, so now we're going to get into the R part inside of our Python notebook, where the %% tells the notebook that now we're coding in R. And this whole big chunk is defining functions that we're going to use for plotting. The library that we use is very powerful, but also not particularly intuitive for beginners. So we're just going to set up these plotting functions for you. And that way you can just tell it which attributes you want to plot and some of the special plotting features that you want to use, like splitting by different variables. So I'll run that. And it should, even though it's a lot of text, it should run really fast. Well, I thought it would run really fast. Yeah, so now mine is done. This is a little scary because it's red, but all of that's fine.
I've noticed when you use a different language, like R here, or if you're using Kale, it tends to output with a pink background, even though it's not an error.
It's a little stressful.
It's stressful, but
Yeah.
it doesn't say anything about an error, it's just telling you what it's doing, and it shows a pink background for some reason.
Yeah, and this is like we were using the extra plotting libraries, and then this grid extra, which is going to help format that. And so basically it went to the server and it went and found all those libraries and downloaded them so we can use them. All right, so now we're actually going to make our plots. And so what we can do is we can explore these different annotations, and especially we can compare which of our true positives have what values of annotations, what values of annotations don't have any true positives, and try and make some thresholds and some decisions and define some boundaries. So the first thing we're going to look at is just the plain qual score. So let's go ahead and plot that. We're going to read in the file first, which goes pretty quick. And this is basically just saying, you know, it's using one of the reading functions from this file. We're going to split on a tab. That's our separator. And then just some extra formatting stuff. All right, so now we're actually going to do our plotting. Well, we're going to define the plot, and then we're going to view the plot. I agree with your notes that we could smush those together. So that is what I would call a long tail distribution. So there's a lot of mass on the left side, and then there's a whole bunch of outliers. So what do we think about those outliers? Are those going to be good variants, bad variants? What do you think? Just from what you know about distributions and outliers. Do you think they're just the cream of the crop, the best of the best? They get the highest scores, so they're most likely to be real? No? Yeah, outliers are weird. They're not like everybody else. They're probably not good things. I can tell you the quality score is effectively the sum of all the alternate allele evidence. And that's why we say we say that the filtering is variant quality score recalibration, because just the qual score itself doesn't tell you whether it's an artifact or not. It just tells you how many alternate reads we saw in all the samples. Those alternate reads could all be errors, but they're still there. They're still supporting the alternate allele. So for the vast majority of sites, you know, they're going to have pretty consistent coverage. And then these outliers here had a lot more alternate allele evidence than everybody else. There are places in the genome where your whole genome has its beautiful 30x coverage, and then all of a sudden you'll see these enormous pileups because there were segmental duplications in your sample that weren't represented in the reference. There are just poorly mapping reads that were ambiguous. But those aren't good regions of the genome, and even though they have a lot of alternate allele evidence, because they're different from everything else, it's a little suspicious. So we don't really like those guys. And I'm going to go ahead and comment out that line. And we'll just look at the big chunk of our distribution here. And then I need to run B again. Cool. That's a more informative distribution. Now we got some interesting things going on. I mean, we zoomed in on most of the mass of the data. That's improvement. It's got a little bimodality, which is kind of funny. There's 2 different peaks there. I don't know what to make of that right now, but if we run this third plotting command, we can actually label these based on how many call sets they were discovered in our truth data. And so now it becomes a little more obvious what some of those peaks are, right? So the things all the way on the left, the lowest quality, they're gray. What do we think about those? Their number of call sets wasn't 1 or 2 or 3 or 4 or 5 6 7. It was missing, so it was 0. So the variance that we're seeing in 0 of the consensus call sets, what do we think, good or bad? Probably not so good. So there's a whole big chunk of things on the left side that we don't believe. They're the lowest quality. We didn't see them anywhere else. And then typically the things that we're seeing in more call sets do have more evidence. They have a higher qual score. So from this plot, we can say the lowest quality stuff, probably not so good. But then once it reaches a certain minimal threshold, then it's kind of hard to say. So we'll use some other annotations too. Should we move on? Okay. So just looking at the qual score didn't really help us too much. I think we got rid of some of the worst variants, but there's probably still a lot of false positives in there. So we're going to start looking at another annotation, and this one's called QD, which is short for qual by depth, or quality divided by depth. And so I'm going to run another one of those functions, and this time we're using QD. And we'll plot this thing, and that's pretty special. So this distribution has a couple peaks. There's one that's on the low side. There's one that's centered around maybe 15, and one that's closer to centered around 30. What do you think those 3 different peaks represent? Yeah, so the ones on the left are definitely bad stuff. So those are the lowest quality, right? Because if we divide the quality by depth, we think the depth is going to be pretty consistent for a genome. Then if those are the lowest values, they have the lowest quality. They're bad, they're gross, we're going to take them out. But the other 2 might be perfectly respectable. So knowing that this is a single sample, what 2 categories might our variance fall into? Well, we in the beginning steps, we did the select variance command to just pull out the SNPs. So we're only looking at SNPs, but that's a good idea. I'll give you a hint. One of the peaks is twice as much as the other one. That's a good hypothesis. You had a really good hint.
Think about the nature of variants themselves. When you are a eukaryotic organism, how would variants differ? How would you see that?
I don't understand the point of how to categorize the variant. I know that the quality of 30 is a good quality, so how I process that information to avoid false positive.
So
What is the point? I don't understand.
So if we know where these distributions come from that make up this big graph, we can justify excluding outliers potentially. So I mean, the question is like, do we want to, like the middle ones usually are pretty good, but the right-hand side, like are those outliers good, are those the best variants or are those the worst variants? Because they're different from the middle, but they're also the highest scores. So the quality score is effectively the sum of the alternate reads. And if you divide by depth, you know, then that's going to normalize it. So for heterozygous variants, you're going to sum up all your alternate reads and the base qualities for your alternate reads. You're going to get 30, 30, 30, 30. And then you're going to divide by your depth, which is about twice as much because you have reference reads also. And you end up with the quality by depth score of 15. And then for your homozygous variant sites, you add up all your base qualities, 30, 30, 30, 30. You divide by the depth, which is all alternate alleles, and then you get 30. So those two peaks there, the one in the middle is going to be heterozygous variants, and the one on the right side is homozygous variants. So in that case, the peak on the right, even though it looks a little asymmetrical and it's like an outlier, then we do want to keep because homozygous variants are good data. In fact, they might even be more confident than the hets. But then the stuff all the way at the end are going to be the really bad allele balance variants, like the 10%, 20% alternate alleles, probably not a real thing.
The peak of the left is maybe error sequencing. We can interpret it like error sequencing in the peak of the left.
It could be, yeah, it could be sequencing errors, it could be mismapping.
To monitoring how is the run of the sequencer or how the library preparation maybe.
There's a,
Maybe it's a sign of that. Not
all
a
the
correct
call
sign,
sets
but
that I've looked at have that, like if they're good quality call sets or if they're bad quality call sets.
Because if I see a larger peak in the lab, maybe I can assume that one reason is that library.
Right, yeah, so if you've been looking at, you know, a bunch of different samples and then all of a sudden you have a sample that has a really high peak, that one might be a little bit lower quality.
Like a little sign that
Yeah, we can probably still clean it up, but it just might be a little lower quality to begin with. So now I'm going to go ahead and color these variants based on the number of call sets that they were supported by in our truth data. And sure enough, the stuff on the left wasn't seen in any call sets. That's bad crap. And then, you know, we see some bimodal distribution, but for the most part, the variants in the middle and the variants on the right had good support from all of our truth data. So again, now the two filters that we're thinking about applying, we want to cut out the lowest qual scores, and we want to cut out QD maybe less than, I guess it depends on how conservative we want to be. I mean, in my experience, QD less than 2 is like the worst of the bad stuff. Less than 4 is still pretty bad. I mean, as you start to get into like 5, now you're going to be here and you're picking up some of this stuff in the yellow and green tails that might be okay. But honestly, yellow and green means 2 and 3 call sets, so that's not so great. But now we can look at multiple annotations together, which can be a little more informative. So we're going to start by looking at QD and depth.
I have a question.
Sure.
Before we keep moving forward.
Yeah.
In the first plot we saw, the first C, we have like
The very first one, the qual?
No, the seed plot, viral seed?
Yes.
For the first one?
Yeah, when it was not colored?
Yeah.
Okay.
We have two pigs. I didn't understand where those two pigs are.
The middle are your heterozygous variants,
Okay.
and the right side are the homozygous variants.
Okay.
Because the quality score is basically the sum of the alternate allele base qualities. So for homozygous variants, you're going to have twice as much evidence, alternate allele evidence, as for the hets. And so they have this one peak that's like twice as much as the other one.
Okay.
Cool. So now we're looking at QD and depth. And we're coloring by call sets again. And we have our one-dimensional graphs, like the QD that we saw before, which is sort of squished on our x-axis here. But we're also adding the depth information, and then we can plot those in 2D. And why, hello, the things that are high depth, sure enough, like all those outliers, the high qual stuff is also high depth. And those are not supported by any of the call sets. Like they're weird. They're not like everybody else. They have too many reads. So we don't really believe those either. So the things that we do believe are going to be these colored points, right? And if you want to do the best job separating your colored points from your not colored points, we can zoom in a little bit. You don't have to, but I will do this while we get everybody caught up. That didn't go as well as I'd hoped. Oh, I think I wanted this one to be 100 and this one to be 20. Cool. So I zoomed in on the edge of our cloud of colored things that are probably our good things that we do believe. Because if we wanted to draw a line to separate things that are definitely bad from things that are looking pretty good, how would we draw that line? And you have a couple different options. You could filter just on QD, and you could throw out this side, and then keep all of this, which is a lot of gray on the top. You could filter by QD and by DP, which would throw out a lot of stuff, but then you'd still have this corner of gray here. You could make an x equals y line, which would leave you with all the stuff here. Or if you could draw any shape that you wanted to do the filtering, what would you do? Yeah, just make a circle for those points, right? Like they're in a nice, beautiful cluster together. Like we don't want to make hard filters. We don't want to draw straight lines. We want to just capture that whole cluster with some kind of ellipse, which is effectively what VQSR does because it's looking at multidimensional Gaussians, right? And so those are going to be, you know, like nice little bell curves in one-dimensional space and nice little hills in two-dimensional space. So that's one of the reasons that we use the underlying model that we do for filtering. So now it's time to actually apply some of our filters and see how well we do. And having looked at all these plots, I'm going to start with qual, which was our first plot. And typically, our best practices say to filter out stuff that's qual less than 30. So those are going to be things that are like less than one good read, like 2 crappy reads, or like 1 not quite so great read. And that's just not enough evidence. So we'll filter those out. And so this was the expression that we want to use to evaluate our filter. It knows to look up the qual field in the VCF and apply that threshold. And then the filter name, qual30, is what's going to get applied to the filter field. So if we look at what comes out, we have a whole lot of pass variance. That's not super exciting. So come on. So if we search specifically for qual30 in our grep command, then we do find that we pull out a whole bunch of variants that are filtered with our qual30 filter. But we saw from the plots that that by itself is probably not enough. So to get the best results from our hard filtering, we're going to want to use pretty much all of the annotations that we have. And now we're looking at 7 annotations. It's not as easy to do this by plotting 2 at a time, but we can start playing with some of the numbers that we have in the best practices. So this is what we recommend as our hard filtering best practices. Qual less than 30 is the one that we just applied. QD less than 2 is conservative based on that 3 peak distribution that we had before. Mapping quality less than 40 is, those are like not your best reads. And then looking at rank sum statistics that are pretty low. So the rank sum annotation is going to compare the values of whatever attribute it is, like mapping quality or read position, between the reference reads and the alternate reads. So in order to have a value, you need to have some of both. Like, so it's only going to happen at sites where you have some heterozygous observations. Basically what you do is you look at the mapping quality for all the refs and you sort them, and you look at the mapping quality for all the alts and you sort them, and then you can calculate a statistic based on how many alternate reads, how many reference reads you have that are better than your alternate reads. And the more disparity there is, the more unevenness between the scores on the reference reads and the scores on the alternate reads, the lower that statistic is going to be. So, for example, like mapping quality, things that have a lot of mismatches will get a worse mapping quality, in which case your reference reads will probably be like beautiful mapping quality 60s, and then the ones with 3 mismatches will be like 50 or maybe 40. If they have soft clips, they might get a lower mapping quality. If they have big insertions or deletions in them, they might get a lower mapping quality. Or if they genuinely don't really look like they map here, like there's another place in the genome that almost looks as good, then they'll get a lower mapping quality. So we want to be able to filter out things that were mismapped or things that were just artifacts altogether. And so we can compare the reference and the alternate reads to do that, because we expect them to have pretty much the same attributes if the alternate reads are good reads. So I'm going to go ahead and apply our best practices filtering recommendations. It's going to take a little while. So then I'm going to take a peek at the file, and I'm going to just look at the things that have a filter status that's not pass. So I'm excluding the headers, and I'm excluding the things that say pass. And so if we look at the filter status, this one's filtered by the SOR filter. This one's filtered by the SOR filter and the QD filter. This one was less than mapping quality 40. So we see a lot of variants are filtered. It's hard to tell what proportion that is, but that they're caught by a bunch of different filters. Like all those annotations are important because all those annotations are catching things. So before we tinker too much, let's just see how our best practices recommendations do. So we'll calculate the concordance. This was one of the variant call set evaluation steps that John talked about, where we look at, well, here we're not looking at genotypes, we're just looking at the variants themselves, the position and the alternate allele. And we're going to do that between our filtered call set and the truth call set. And we're going to do it only over these high confidence regions. So there are parts of the genome that are messy and hard. And in those places, the callers didn't have a lot of consensus. So we excluded those from the truth region. So the things where they had, all 7 callers or 6 or maybe even 5 callers that were in agreement, we're pretty confident in those. If none of the callers called it, we're pretty confident in those reference bases too. If there was a lot of discrepancy, we excluded it from the confidence intervals. So it might be a real thing, it might not, but we're not going to use it to evaluate. Questions? So I'll run this. And here we're evaluating just the file where we did the filtering for qual less than 30. So only using the information from our original plot. And then this file is the one using all of our 7 filters on 7 different attributes. And we'll calculate the scores for those. It's still running.
One thing I don't think we've mentioned yet, but it's important to note, filtering doesn't delete the variants from your file. You'll notice that we've been continuously pulling them up. It just marks them with something in the filter field. And if a tool in GATK sees anything that's past there, it doesn't use that in its data calculations. So they're still there. You can recover them if you later decide, hey, my filtering was too stringent. I need that variant back.
And that's what a lot of our analysts will do in the Mendelian disease space, is they'll look at all the passing variants, they'll check them out, they'll run their functional annotation, and then they'll say, gosh, I didn't have any good candidate variants at all. Okay, let me look at all of the variants, even the ones that failed filters, and then see if I find something there. So you kind of go through your first iteration of your analysis using just the best variants, and then if you still don't find something, then you can look at the other ones because they're still in the file. You just change the filter status. Well, this is effectively what we're doing now. We have the 2 different VCFs that had 2 different filters, one of which was just the qual less than 30, and the other one was all 7 of our best practices filtering. And so we'll look at what the concordance actually is for those 2. And again, we see zeros for indels because we excluded them here. This is not bad. This is respectable. The sensitivity or the recall is pretty good, 99.6. Go team. The precision is also pretty good, but in theory, our best practices filtering scheme should be even better, right? So what happened here is we got stricter with our filters. So the false positives fell from 387 to 99, but at the same time, the false negatives, the variants that we filtered that we shouldn't have filtered, increase from 244 to over 1,000. So on the one hand, we got rid of more false positives, but we lost some real things at the same time. And for hard filtering, a lot of the time, that's just a trade-off that you have to make. And depending on what kind of analysis you're doing, if you just want absolutely high confidence variants because you're going to have to go pay for Sanger sequencing on all of them, then you want highest precision possible. But if you're looking for that one causal variant in some sort of rare disease, then you're going to go for higher sensitivity. And if there's some false positives, then you just figure out how to deal with that.
Laura, if I have a case with this data that I'm sure that the disease is a Mendelian disease, maybe it's a SNP, and I'm very sure of the region of the genome that is related with the disease of eating in the case. So maybe this approach is better because they try to eliminate all the trash and all this stuff and try to convince me that this recall is better because I know the specific mutation that I'm looking for, or it's not good enough and try to not cut off maybe some other mutation in the same gene because we eliminated it and maybe it's related with the disease. I don't know if
I think I get what you're saying. So as you do, depending on the type of analysis you do, there are additional filtering methods that might be available. Like if you do your functional annotation and then you filter down to just things that are damaging, just things that are missense, loss of function, or if you have a candidate gene list for the particular disease that you're doing, then you filter down to just areas of the genome that have been shown to be associated with the phenotype that you're studying. And in that case, if you have another filtering criterion, you can be a little more lenient because those false positives will get taken care of by the functional annotation or because they're outside your regions of interest. So you might be able to use other tools depending on the information that you have. Yes?
Looking at the performance matrix, actually you see fluctuation between the number of false negatives in one and the other. And that's always an issue in human genetics. Because, well, you rather have, you never want false positives. Because if you're dealing with false positives, then that's actually messy. Because, you're telling someone you're okay down the line, and then they're not. But you see a fluctuation from, like, you have your false negatives actually, but going back to the point, your false positives dramatically increase. You have in one instance 244, and then it goes up to almost 1100. Is this kind of like reading tea leaves? Like you have to play, like you have to play around with your parameters and the filters until you kind of like decrease the. And I have no issue whatsoever if the answer is yes, because some of these things are actually like reading tea leaves. So if the answer is like you have to find the right combination of filters, then I'm totally fine with that.
So, like I said, there are 3 different filtering strategies. There's hard filters, there's CNN, and there's VQSR. CNN and VQSR are kind of the state of the art. CNN we use only for single sample, but if you did only have a single sample of data for whatever reason, I would recommend that over this approach. We'll play with that after the lunch break. And then if you have a large cohort, then we would recommend using VQSR. But if you're stuck doing just hard filtering, then yeah, it's going to be a little hard. The recommendations that we have are very conservative. I mean, if you think you're a bioinformaticist, you can understand this in a little more detail. The rank sums, those are z-scores. So if you think about a z-score that's less than -8, that's like the worst of the worst of the worst stuff. Like that filter is probably not doing very much actually, because it's only filtering out the worst crap. The FS is a Fisher exact score on the strand contingency table, and I think it's a Fred scaled p-value. So like, 1 in a million, if it's like worse than a 1 in a million chance that it's actually strand bias, but you just happen to get some good observations, like that's pretty conservative too. So I mean, with a little more domain knowledge, you can kind of apply thresholds that might make more sense numerically based on what your goal is. We set these because we wanted to be conservative and we didn't want to overfit to any particular data set that we looked at. But yeah, if you go with the hard filtering approach because you're working on a gene panel and you don't have enough territory for VQSR, or you're working in a non-human organism and there aren't any resources to do the VQSR training or the CNN training. Actually, we have a script to train new CNN models, so that would be an option. But if for whatever reason, like your experimental design precludes you from using the other 2 methods, this is what you get, and it's a little bit like tea leaf reading, and it's a little bit like, trying to stretch a blanket to cover a small bed or whatever, and because you can't, there's pros and cons. Like as you start to change these filters, like you will gain, true positives and lose false positives. I mean, when John talked about filtering, he talked about the precision recall curve. And I mean, you want your precision recall curve to be like a beautiful right angle so that as you add more true positives, you don't see a big increase in false positives. But in reality, if on a method like this is going to have like a really crappy little precision recall curve, yeah, it might even be like below x equals y.
Yeah.
In which case, the value that you get as you start to include more true positives might introduce proportionally more false positives, and that's just not worth it. So unfortunately, it just does depend on what your application is, and you got to make some calls.
Actually, I appreciate the answer because I understand that underestimation might be like the way to go if you actually want to generalize the way, the applicability of the pipeline or the workflow. So that totally makes sense. Looking at, if you actually want to use it for multiple approaches and be capable of actually using it for many, or generalize it, actually, that's the correct word, making a generalizable approach totally makes sense. Looking at also one of the goals of what we're doing right now.
So on this note, we actually have a take-home exercise kind of structured around this. It's involving this notebook. So you see this, all these filters you're doing here. You can take every single one of those values that we're filtering on and go back up to C or D, I think it was. The last graph.
Yeah.
Try plotting the different annotations against each other and look at the graph and see where you might want to draw a line. And then you can put those lines that you're drawing, which are your hard filters. So for this one, maybe you want a QD of 8. You make your filter be QD less than 8. And you can run the filtering again. I'd recommend outputting it to a different name so that you don't overwrite it. And then you can run concordance at the end and see if it's improved your recall and improved your precision. We used to do this in the workshop, but it's kind of like an all-day thing. People make teams and try and improve their scores.
That's fun.
So we don't have time to do it today, but if your studies tend to do hard filtering, I would really recommend going through that exercise and really take our starting point of filters and really fit them to this sample and see what you might want to fit, what filters might make more sense. See if you can beat what we've come up with.
Yeah, anecdotally, I can tell you that there are a lot of variants in that truth set that are low mapping quality. And they're low mapping quality because there are multiple SNPs within the length of the same read. And BWA says, I couldn't find a better place to put this, but I'm still not convinced that this came from this part of the genome. And all the call sets agreed, so we mark it as a true positive. Maybe it's not a true positive. Maybe it's a segmental duplication that diverged from the reference. They have pretty good allele balance for the most part. But there are things in here, true positives, that were missing because of that mapping quality filter. So if you really wanted to get the best sensitivity possible, like I know that you'll have to roll that one back. But. So you can tinker with that, and then after the lunch break, we will run the CNN, which is a lot more automated, but also a lot more complicated under the hood.
There's no more questions?
For lunch, we did our basic hard filtering, which is sort of the vanilla flavor. You know, it's a lot of manual effort. Maybe the results are pretty good if you put some time into it. Now we're going to do the other single sample filtering option, which is the convolutional neural network, which is one of the things that John talked about. And this is a lot more automated, but also a lot more complicated. So we're going to use the same runtime values yet again. If your notebook is up and running, then you're good to go. Yada, yada, Python 3. and we will download the files for this notebook. Just gonna shift enter, shift enter, shift enter. This is not the most fun part. All right, so my files are almost there, but there's two different flavors of the neural network. There's the 1D and the 2D, and we refer to them that way because it's the one-dimensional flavor and the two-dimensional flavor, where the one dimension is the reference sequence. John showed you all those cool motifs that the model learns, like the homopolymers and the TG-TG repeats, and all that is just based on that linear sequence of the reference. It also has a little perceptron to use all the annotations that we looked at before in the hard filtering tutorial, and the annotations that are also used by VQSR. The 2D model we're not going to walk through today. We'll leave that as an exercise for you to play with at home. But that one is 2 dimensions because it's you can kind of think of the X dimension as still being the reference sequence, but now it also has all of the reads with it as well. And so it's effectively getting as input what you're looking at in IGV when you're trying to evaluate variance yourself. It's looking at all that read data, and it's got additional channels in the tensor to give you forward strand, reverse strand, and insertions and deletions get their own channel, and so on and so forth, which is a lot more data, and so it runs more slowly, which is the main reason that we don't want to do it today together as a hands-on exercise. But it's a little bit more accurate, and it just kind of feels better to actually use your read data. Yeah.
Would it be better to use GPUs for that one? Or just a regular CPU would be like for processing?
Yes, so to run the 2D CNN with a GPU would be better, but right now, as of today, we can't access GPUs from inside the Terra notebooks.
Yeah,
But if you were running it like on your own virtual machine, then definitely.
Okay.
Yeah. I think from what Sam told me, the speed up is kind of proportional to the cost difference on Google Cloud. But I mean, at the same time, like having your results faster, like can you really put a price on that?
No, you can't.
No, you can't.
No. I mean, if you have GPU access, it really makes a difference if you're using convolutional neural networks. So, yeah, I mean, it makes sense.
Yeah, I mean, somebody mentioned Colab, like you could probably run one in Colab.
Yeah, we were talking about Colab yesterday.
Yeah.
You can also get like, I think it's 12 gigabytes RAM on a GPU for free at least or something.
It
You
is
can put on a
free.
queue or something, so but I mean, can you put a price in free? No.
All right, so I have my data, hopefully you have your data, and we're going to run this initial CNN score variance command. We're using a slightly different VCF here. It's still an NA12878 VCF. You'll see from the file name, it's on the B37 reference, which doesn't really matter because we have all of our data for HD38 and B37 rolled into one training model that's built into the GATK with this command. And this is 1 megabase of chromosome 20. So it's actually a smaller amount of territory than the variance that we were looking at in the previous exercise. Again, because we want to be able to do this in a reasonable amount of time while we're doing it live, walking through together. But you know, it is fewer variants. The results are going to be a little bit noisier. But at least we can see how it works in action. So we'll run that. So it gets a -r argument with the reference, which it's going to use as the context to evaluate the model. It's going to get the annotations out of that VCF. And it does take a little bit of time. So basically what's going to happen is it's going to generate this aggregate score based on all this information. So for example, VQSR has all these different clusters of Gaussians, and it'll generate its own VQSR score based on the values of those annotations evaluated against the different clusters of Gaussians. This one is going to generate its own single scalar score for each variant based on the annotations and the reference score once it does all its convolutional magic. But the idea was to make the interface and the output very similar to VQSR. So we have the same idea of putting in a VCF with the same input, assigning it a score field in the info field, and being able to set the filtering threshold in the same way. And mine is still going because there's a lot of neurons, a lot of them. I mean, if you have any experience in signal processing, the convolution is effectively like taking this matrix kernel and then applying it to each window on the reference. So each layer is like many, many, many matrix multiplies, which is a lot of math compared to what we were doing in the previous exercise, which is just like, is this more or less than 30? Okay, let's filter it. So this is why it takes even longer on even less data. But it is significantly more accurate and a lot less effort from you because the computer's doing all the effort. So now my command is done. I should have my scores applied to my variants. And just like VQSR, it labels them with positive train site where applicable. So it'll use the same resources like the Omni 1000 genomes genotyping data, the HapMap variants. DBSNP doesn't actually get used. It's just sort of for plotting purposes. And was it Axiom? Yeah, I think we have genotyping data from one other type of array. But all of those are our high confidence common variants. So if we see one of those in our data set, it's probably a real thing. And then we'll label them in the output VCF. So that's to say that this variant is a positive training site. It got used to help build the model. This VCF has already had VQSR run on it, so it has those VQSR scores applied. But we'll see that we also have our own CNN 1D scores. The scores between the two models aren't really comparable. The scores between, I don't know if it's true for the CNN. Certainly for VQSR, the score distribution for one call set is going to be totally different from another call set. But like, roughly speaking, positive scores are better, negative scores are worse, and bigger becomes more extreme. So the fact that this is a positive training site, we've seen it before in our high confidence resources, and it gets a positive score, that's good. The model is working as we expect it to. The CNN should update this culprit field, which will give you the annotation that looked the worst, effectively. So, I mean, out of all the 7 annotations, maybe some of them look good, but some of them don't. Although, in this case, the mapping quality is still 60, so that's a little suspicious. But for the one below that, it's a little weird too. Maybe these values are not actually accurate. Maybe it didn't update the positive train site. But just to show that we've run the scoring tool, and now we have all these CNN 1D values for each variant. And then we're going to apply the filters. I don't know why this didn't stick. Were there 2 different places we fixed this, Kate? Well, fine. So the thing. Yeah, I'll change mine. So when we talked about the CNN earlier, and someone said, oh, well, it's just hard filtering on a different number. It's still not. You're not the person deciding the threshold. You tell it what sensitivity to common variance you want to achieve, and it will pick the threshold. So you supply the filter variant tranches tool with 1, 2, however many resources you have of very, very high confident variance. And you tell it what sensitivity you want to achieve with respect to those variants. So We typically have better performance on confident things, or on common things, so we want to have a very high sensitivity. So these values actually are not going to give us as good results, actually, even as the hard filters, unless we increase them. So we're going to say that we want 99.9% sensitivity to common variance. And actually, when we do the concordance evaluation at the end, we'll see how approximate that number actually is. So what happens is the tool evaluates all of the variance in those files. It counts them up, it marks them for the sample that we have, and then it finds the threshold of that CNN 1D score, like given that they're all sorted and ranked, it finds the threshold where we have captured 99.9% of those variants. And so that way you don't say, okay, I want everything with a score greater than 3, because that doesn't mean anything to you, but you can tell it if you want high sensitivity, or maybe you turn the sensitivity down because you want higher specificity. But you can change that parameter in a way that's a lot more intuitive. So I'm going to run this with my new 99.9 parameters. Also note that because that was a VQSR filtered VCF, we're going to run with this invalidate previous filters argument. Because otherwise it'll only apply the CNN filters on the things that VQSR thought were pass, and we won't be able to recover anything where VQSR was wrong. All right, so we are going to look at how we did. And we'll run that concordance tool again. So in theory, this should be over the confident regions as well. And we're going to output that concordance table. And this says unfiltered VCF, but we know from looking at the output up here, it has VQSLOD filters. It had filters applied to it. It said pass before we did our own filtering. So that file by itself is the, change this again. It's the VQSR filtered VCF. So we'll run that. Can do. This is also taking longer than I expect. I'm very impatient. All right, so let's see what the concordance was for the original VQSR filtering. Not bad. The sensitivity is pretty low. In this particular case, we're looking at SNPs and indels, whereas in our hard filtering, we were dealing with SNPs separately, but the CNN model can handle both at the same time. So indel sensitivity is actually really quite good. The indel precision is not good, which I've seen before. I haven't seen it quite that low, but again, this is on about 10,000 SNPs and like 2,500 indels, so it's not a big sample. If you think about 4 million SNPs in a whole genome, then that's a really, really small sample. So it's going to be noisy. But we can compare that to what we have for the CNN model that we just ran. And you'll see we specified 99.9% sensitivity, and for SNPs, we were really quite close. And our sensitivity is better than it was for VQSR. Our precision's a little bit worse, but you know, pros and cons. So I don't know, is 1,400 false positives a number we can live with? Maybe. Again, it's hard to say over such a small percentage of the genome. Compared to VQSR, now we're doing a much, much better job in indel sensitivity, and the precision is about the same. But again, it's a small segment of the genome, and we also didn't run with the confident regions, which I'm a little concerned about. but we could change that. Do we have time to change that? So, I mean, and we can play with the parameters a little bit here. So if we want to change, so I told you 99.9 was probably pretty good, but if we go back to 95 that we had before, we can see how those values change. I'm going to let that run. And I'm going to go get our confidence regions, except that we didn't download them. And so now I've set a lower sensitivity for both SNPs and indels. I expect that the sensitivity will be lower, but ideally the precision should be better too. And then we will rerun our concordance. And if we output that again, how does it change? So you see now the sensitivity is much lower. SNP sits about 92%, indels it's 91%, but the precision now is looking better than it was for VQSR. So again, like there's always going to be a compromise between false positives and false negatives. Ideally, for a model that's performing really well, the variance should be ranked in such a way that all your true positives are at the beginning and all your false positives are at the end, and so you can add more true positives without adding false positives. But how accurate your model is going to depend on how well you rank them in that way. So I think we could get a more accurate estimate actually if we included the confidence regions for the NIST genomes in a bottle section, but again, it is a very small slice of the genome. I can tell you, I myself ran the data that we had from the hard filtering exercise with just the SNPs. I had to modify the code because there was a bug where it wouldn't run without indels, but it did actually do better than the hard filtering on SNPs. So if you're interested, you can tinker with that. You can also take that VCF and actually revert the filters to do a comparison versus unfiltered rather than a comparison versus VQSR. So, and then, of course, there's the 2D model, which is, it's going to take a little while, you know, get it running, go make a sandwich, come back. But it is a little bit more accurate. These numbers will probably have to change too. So
we change those back to the ones we had before.
the 95 and the 99?
Yeah, exactly.
Yeah, so I don't know where the 95, yeah, I would suggest that you change these to 99s as we did for the 1D model as well, because this really will limit your sensitivity to less than 95%, and we can do better. But yeah, it's just to show that there is this parameter that you can change and that it'll affect your sensitivity, but also specificity as well. Is there anything else? Yeah, so this is our gold standard for single sample filtering. We're going to put it into our clinical germline whole genome pipeline. It's, how much does it cost per genome? It's not an insignificant amount.
If it's a featured workspace, you can click on that workspace and it'll tell you.
It is a featured workspace.
Was it in here?
Oh, because it's just notebooks?
Yeah.
It doesn't have the workflows?
Do we have a workspace for CNN?
Yeah.
If you go to the hamburger menu in the top left, and then go to library, then showcase. Should be able to find it. It'll be on the left side, I think.
Yeah.
And then it'll be on the dashboard. There you go.
Oh boy. This must be the 2D. Because that's astronomical.
$13?
Yeah. I know the 1D's not that high. So we try to put cost estimates on the featured workspaces to go with the workflows that they have. We are going to revisit this one because $13 for a single sample is not great. The example that we have that's just the truncated 20 kilobase BAM is 4 cents. So that's on par with what we've just run. But we are going to investigate that cost estimate because I thought that the 1D was like in the neighborhood of $1 per sample. The 2D is admittedly more, but that seems really high. But again, like we talked about yesterday, like how much money are you going to spend? How do you know how much money you're going to spend? We do try and give estimates for our best practices workflow, and we will revisit this one to assess its accuracy.
Now, if you scroll up, it tells you what workload it's talking about. So that's
It
the
talks about
1D
both.
and 2D.
Yeah.
And then this optional one should also give you a cost.
Oh, that's the training one. Yeah. So if you have different data, or maybe like John, you're working with long read data, and we know that the CNN model was trained on Illumina short reads, although from different Illumina sequencers. And you want to train a new model for whatever reason, we do provide a workflow to do that, where you're going to need some truth variance and some confidence regions. But you have the capability of making your own CNN model for different data. And then in the CNN workspace, there's also this HAPPY evaluation workflow, where HAPPY is hap.py for Python. And that'll give you some nice plots for the sensitivity and specificity, and it'll give you the precision recall curve, which is nice to visualize like how changing that filtering threshold is gonna balance between false positives and true positives. So if you're excited about the CNN, there's more to explore in the CNN-specific featured workspace.
Any other questions?
All right, then we're gonna move on to genome-wide association studies.
While I'm getting set up, I want you guys to do something, and that is delete your cluster. I know. We're actually going to replace our runtime with a new runtime, and in order to get the settings for that, so you should be probably here, you're going to open up the hamburger menu, go down to Library, and then pick Showcase. So that's going to take you to our list of featured workspaces. The one we're interested in is going to be in this rightmost column, a little bit of the ways down. It's called 2019 ASHG Reproducible GWAS. So click on this one here. We're going to clone it just like we did before with the last workspace. So since this is read-only, meaning you don't have access to edit it, we're going to go 3 dots in the upper right corner and then select Clone. When you do, this menu is going to pop up here, and you're going to just replace copy with whatever identifier you probably used for the last one, but some sort of identifiable name to you. For billing project, you're going to select, again, your free credits account, and go ahead and click Clone Workspace. And then I think you had to use Workshop Temp, not your free credits, so use Workshop Temp again. So once we clone the workspace, so in the new workspace, you'll notice that your runtime is still running. Your runtime is universal across the whole billing project for you. So even though I'm in a different workspace, I'm still using the same runtime environment, which is great because it means you can take your files from one workspace to another inside that runtime. But for our purposes, we actually need some different runtime settings. So those runtime settings are way down here. I'm gonna scroll back up to the top and go through entering them to show you it kind of in place a bit easier. But you'll go to update runtime. The environment we want to choose is Hail. Our profile should still be custom, but the CPUs we want is 8. And we actually don't want GATK on this one, so we're going to delete that startup script. Next, you check Configure as Spark Cluster, and that opens up a whole nother menu. You'll change this workers to 4, and that's it. So this is what everything filled out should look like. You can go ahead and hit Replace once you have it. So just to walk it through one more time, what I changed was the environment should say Hail now. Your CPUs should say 8. Your startup script should be blank, so delete the text that's already in there. I checked this Configure as Spark Cluster, and I changed workers to 4. Then we're just going to replace. It'll give you a warning about how it's deleting the files, but since we're doing a completely different analysis, it doesn't matter that we keep those. So we're going to go ahead and hit Replace, and it should start creating. Yep.
Number of workers?
Yeah, the number of workers.
Here. Pull this up here. So the number of workers is 4.
Okay. 4 workers. Oh, it's $1.16 an hour.
Yes, this one's a bit pricier. It's because we're running with a Spark cluster, so it's got more to it. Were you the one asking the question about the GPUs and notebooks?
Mm-hmm.
I did actually ask the notebooks team about that. They have a ticket for it. They've been actively working on it. I don't know when it's going out. I asked. I'm waiting to hear back on that part, but it's coming.
Yeah, no,
So.
that's actually good stuff.
Yeah.
Well, one of the issues, we were talking about it a while ago about Google Colab and GPUs is actually that it depends on GPU availability.
Yeah.
So there's actually a queue for GPUs. So
Mhm.
it's still the same thing, like unless you want to, you've got the money to pay,
Yeah.
there's GPU availability, like that actually works.
Yeah.
But if you don't, if you're willing to wait, and also you can get assigned to a different queue, and depending on that queue,
Mm-hmm.
I mean, you can end up getting not the optimum availability for what your task requires.
Yeah. Yeah. But you can use GPUs in workflows. You just can't use them in notebooks
Yeah.
yet. Yeah.
No, that makes sense. I mean, can you parallelize, or is there the possibility of parallelizing tasks
In notebooks?
Yeah.
Yeah, so that's what Spark is actually doing. It's doing parallelization.
But if you're gonna, well, yeah, if you're calling on Spark, but if you're like kind of doing your harnessing your like local work, like I know that Python has the, what is it, multi-thread, I think it's called,
Yeah.
the library, or multi-process, yeah, and you call like, you do it in a loop,
Yeah.
And you call it like proc, and then inside you build all the arguments. Can you do the same thing locally?
I'm not sure. I'd have to ask. I've never tried it.
Because we sometimes do like image processing, and it takes a long time to do it locally.
Yeah.
Because it does it one at a time. It queues everything one at a time.
Yeah.
And you can do it like, you can
Is
multi-process.
it something you could do with a workflow? Like, is it an automated process, or does it need you to do something
Oh, well, I mean,
interactive?
well, it's not, like, we don't have it as assigned as a workflow per se right now. We're just like in initial stages. We just have like, the first stage is actually like inputting images. It's a defined function. We created our own custom function to actually import all sorts of data, like either images or CSVs or whatever. And then it recognizes it, it creates a tensor, and then the tensor can be processed for any deep learning task as we want, classification or regression.
I think that sounds like something that would work better in a workflow. If you were able to convert what script and tools you had to a WDL workflow, which I'm hoping will be easy because you say it's written in Python. Yeah, you can just plug Python code directly into WDLs and be able to do it that way. Then you're able to just hit launch analysis and it'll run it on everything in parallel at once.
Okay.
Yeah, I think there might be, and then you can use your multi-threading options that you already use in there, and then it'll also be able to launch on like multiple images at once if you need to do that. I think that might be a good option, but we can ask about multi-threading in notebooks.
Oh, that's great. I appreciate it.
All right, so my notebook is running. Is everybody else seeing running for their notebooks? Awesome. Okay, so before I get straight into the notebook itself, I wanted to set some context. So as you noticed from my deleting GATK from that startup script, we're not actually using GATK in this notebook, but this could be a realistic step in a study. So after you use GATK to call your variants, then you might want to do a GWAS. So first up, we're going to take a look at the data, and then we're going to explore it in a notebook, and then at the end, we're going to run a workflow on the results from the notebook. So we're touching everything today in the workspace. Tabbing over to the data here, I've shown you this tab before, but in our last workspace, it was empty. So this one has a bunch of different tables set up. The first one here is the sample data table. It's got 2,504 samples in it, which means it's got 2,504 lines or rows. There's multiple pages you can go across to look at them all. But if you slowly scroll this way, you can see all the different columns that are set up. And you may recognize this as metadata. It's phenotypic data about these samples. It's giving LDL, HDL, height, population, where it's come from, the gender, total cholesterol, things like that. So, can't swipe that way on a Mac. All right, so this is a phenotype data table that's set up for our samples, but it doesn't have any of our VCFs in it. This next one here is a sample set, and this is sort of a shortcut if you want to skip the notebook, because this is one of the outputs of the notebook. If you want to skip the notebook and go directly to the workflow, you can. But we're going to be creating a set today in the notebook that's going to have a bunch of phenotypes, and we're going to decide what outcomes and what covariates we want to track. The other one down here is in workspace data. So this is a list of VCF files and their paired GDS files. Let's see, hit edit, do I see it? Yeah, that one's, it's kind of hard to read in this format, but each of those links is linking you to a file that exists in a bucket, I believe. Yeah, if you click on it, you can see the file name. You have the option to download it if you want, but I don't really recommend it right now. And the reason we have this list of VCFs and their paired GDSs is that the workspace we're going to be using takes in GDS files, so we've generated a matched list of it. I'll talk about that a little bit more later, but I just wanted to familiarize you with the fact that we have VCFs and we have our phenotype data. The last bit is our files. So this is our Google bucket. It's empty right now. We're going to be generating some stuff to put in there. So going over to our notebook, you'll see that there are two notebooks. We have this GWAS initial analysis cleared and the one that's completed. If at any point you get lost or you just want to follow along without clicking anything, feel free to go over to the completed one. It's just something that's had every single step run in it so you can see what the outputs look like. But the cleared one is what I'm going to be walking you through today. So I've just clicked on that cleared notebook and I'm going to choose Edit. It's going to take a minute to copy over the data. All right, so one quick thing, I wanted to give a little disclaimer. The data in this workspace it's real data, but it's fake phenotypes. So what we've done is we've modeled the results, the phenotypes, so that when we run this through, it'll come up with some real associations that have been seen in papers, but it's not real in the sense that these are actual samples that have actually been phenotyped and given that information. We do that so that we can share this information with you because of various different privacy laws. We want to make sure that this is a publicly accessible set of data that we can use for training purposes. So while this data will be useful to show you as a training example, I wouldn't take this data and add it to your study because it's not real. The VCFs themselves, though, are real VCFs that have been taken from the 1,000 Genomes Project, so that's all publicly accessible data. Alrighty, setting up our notebooks. So you guys should be pretty familiar with setting up notebooks. I'm going to kind of skim through some of these. We've already set up our runtime values, but it's there in case you need it. We're going to check our kernel, which should be Python 3. Fun fact, the environment we chose, which is this Hail environment here, if I go to kernel and change kernel, it only has one available. So depending on what environment you choose, it tells you what kernels you have options to pick. This particular environment is only set up to run with Python because Hail is only set up to run with Python, I guess. So we've got that set. The next one, we have a notebook extension we're going to be using in this notebook, and we're going to be adding it to our other notebooks. We just haven't had the chance yet. It's something called code folding. So what I want you to do is run this cell block, and when it's finished running, just go to the top of your browser and click this reload the page button. That won't delete your cluster or anything like that. It won't delete any of your progress. It's just refreshing the page so that it can re-render some things. And what we did with these notebook extensions is it allows us to collapse parts of our code or maybe parts of the text that may be bonus information that you don't necessarily have to know. But if you're curious, you can open it up and look. So our first example of that is actually in this very next section, this load Python packages. You'll see at this line you have capture written and then this double arrow here. That double arrow means that something is collapsed with our code folding. So all you have to do is click on it and it'll expand it like that. And then it shows you a whole bunch of code that we're running in this cell. So if you're interested to know what particular packages we're importing or you want to run the same type of code in your own notebook, you can open these up and look, but it's not necessary that you have to look at them. We find that people who may not be as familiar with code tend to get pretty overwhelmed by large blocks of it, like that R script in that last notebook. You guys didn't need to know what was in it really, because it was written for you. You don't need to edit it, you don't need to interact with it at all. But we do need it so that we had those nice functions to generate the graphs.
So you're going to see some examples of that throughout the workspace. If at any point you want to expand the code and take a look at it, feel free. I'm not going to. So in this particular cell, we've loaded some Python packages. There are 4 different types that we've loaded. The first one is called FIS, which stands for FireCloud Service Selector. You might hear the word FireCloud a lot when we talk about Terra. FireCloud is the predecessor to Terra. So first we had that name, and then we redesigned the whole interface and everything, and we renamed it to Terra. So some tools that access FireCloud still access Terra in the same way, and so they kept the name. So FireCloud Service Selector. Basically, it's going to enable us to access some APIs and do some Python code. The next one is Pandas and NumPy. These are both packages we're going to be using for data analysis. We've loaded up pprint for pretty printing, and then matplotlib and seaborn are the 2 libraries we're going to be using for plotting. All right, so let's take a look at these phenotypes. So next thing we're going to do is set our environment variables. And this threw an error, of course. Sorry. I tested these last week and they changed the hail environment yesterday. Just a moment. Okay. Add this line at the top to import OS. It looks like the runtime isn't by default loading OS anymore, so we're going to add that line in. All right. So with that change, is it working for everyone else? Import OS. It should print out this text down here at the bottom telling you what your billing project is, what your workspace is, and what your workspace storage bucket is. And that's just verifying that we've captured those environment variables. If you don't see this, raise your hand. Someone can come around and help you. All right. The next one we're going to do is use a read CSV command from the pandas library to read in a comma separated value that we're going to use FIS, which is the FireCloud Service Selector. We're going to use that tool to grab the data table from our data tab. So what this command is doing is it's going to the data tab in our workspace and it's grabbing that samples table that we just looked at. So go ahead and run the cell. Why? Katie's not defined. Oh, I didn't run this. Oh, that's why. That's why I wasn't importing the OS. I was so busy talking about it, I didn't run it. All right, there we go. Is everyone else seeing a table like this? Seeing some nods? Great. All right, so if you remember from our data tab, this was what the table looked like. Has all these different columns up here, and we're showing the first 5 samples. If you scroll up here, we have a whole list of what the column headers mean. In case you're curious about various abbreviations like FI, fasting insulin. You always have that to reference. All right. So next, now that we've loaded in our samples, we're going to take a look at the phenotypes. So our goals in this section, we're going to use some more plotting functions to visualize the distributions of our phenotypes across our samples. So we want to look for these correlations so we can filter out these relationships in order to prevent ourselves from making conclusions based on something that shouldn't be included. So when we generate these plots, I want you to think about what should these traits look like? What do we expect from these distributions based on what the trait is we're plotting? So the first step is to run some code in this cell, code for plotting functions. This is a big script that, again, is loading up some nice, more easily usable plotting functions for you, so you don't have to interact directly with a lot of these oftentimes complex libraries. And what these plotting functions do is give you these three options for plots. The first option at the top is great for univariate distributions. So you'll see that I'm only plotting one variable here, which is our LDL column from our data table. And we're plotting it, and it's generating both a histogram and density plot, which is the line on top of it, to show you the distribution of how LDL is seen in this population. When we look at this plot, we kind of expect it to be in a nice bell curve, so this particular data makes sense for that one. This univariate distribution using this function kdplot is a good one to use for any continuously valued variable. So you'd want to use it for something like this where you expect to see a nice bell curve. It's not as good for something where you expect to see like a point here, a point there, where it's not continuously valued. The next option for continuously valued variables is the bivariate distribution. And we did a very similar plot to this with our variant filtering, where we saw the histograms on the side, our density plots on the side, and the scatter plot in the middle. So we use this function called bivariate distribution plot for that one, and we feed in two different variables. So in this case, we're looking at HDL and WHR, which if we scroll up are the high density lipoprotein and the waist to hip ratio. So we're seeing if there's any correlation between those two variables. And we see that we have this nice scatter plot in the middle, and by seeing a Gaussian distribution there, that's sort of normal looking data, and that's a thing we can look for to explore it. The third one here is box plots. They can be used for relationships between continuously valued variables, like we've been seeing so far, and categorical variables. So in this case, we're looking at height across ancestry. So you've got these box plots that show the differences broken up additionally between the different sexes, so male and female, and the various ancestries for it. So that's what that particular command looks like. Now, the next three sections here are giving you a chance to play with those. So I've got the code for the one that we just talked through and rendered, but I want you guys to start exploring. Look at different variables, different options you have for it. I will say the text says that for bivariate distribution, you can use a different kind, not scatter. Only use scatter for this purpose. We're just looking at that part of it. This particular table isn't set up to work with the other types of things that this particular function could do. So just change out these variables. right, so let's have a volunteer. Who wants to tell me what they plotted for this univariate distribution? What variable did you choose? Anyone? HDL? Okay. Let's see what that looks like. All right. And based on this distribution, does it look like a nice bell curve to you, or is there something? Yeah, so there might be something going on there. Is there another variable that someone plotted for univariate distribution? What variable did you choose? FG? Okay, so I think FG is fasting glucose. And what do you think of that distribution?
It's pretty normal.
Yeah, it's a nice bell curve. All right, bivariate distributions. So we need two variables for this. Someone shout out a variable. What variable should we put in the first spot? Okay, we got FI. And what's the other one? What? BMI. So fasting insulin versus BMI. So that one's got a nice Gaussian distribution as well. We're seeing some good bell curves on the sides, and then a nice Gaussian distribution for the scatter plot. All right, and then for box plots, I played with this a little bit at home. I couldn't find another categorical variable to play with, but I didn't pick everything. So let's think of what continuous variable we can track across different ancestry populations. We've got a whole list up there. Someone shout one out. TC. All right. So with that one, let me double check what that one is. Total cholesterol. All right. So we're looking at total cholesterol across different ancestries. So we see kind of a dip with the South Asian, I think, is that one. And then maybe African is a little bit higher. So maybe there's some differences in the way those populations were sampled that might lead to the difference in total cholesterol. All right. Are we happy exploring the data like this? Yeah? Had a chance to do it? So the reason why we like to query these things with plots in this way is because it can really inform you if there's anything strange going on in your plot. So I like to give this example that a coworker gave me. There's a cohort of data from a group called AMP-PD, which stands for Accelerating Medicine Precision for Parkinson's disease. So they're studying Parkinson's disease data. And they thought they had found a correlation between a certain medication and the effectiveness of that medication. But when querying the data with plots like these, they found that there was something odd going on. And it turns out there was an inconsistency in the reported start date of medication. So I think it was something like some people were saying the start date of when they started taking the medication, and some people said the start date of when they saw the doctor and were told to start taking the medication. And these underlying relationships of things can skew results and end up with finding that a medication is effective when it really wasn't. So it's really important to do GWAS things like this to query that out. So now that we've taken a look at our phenotype data, we're a little bit more familiar with that. We're going to work with the genotype data using something called HALE. Now HALE is built on distributed computing, and it can essentially be thought of as a way to parallelize operations. So rather than running one operation at a time in sequence, you can run multiple at a time in parallel. And in that way, you can decrease what they like to call wall clock time. So even though you have like 6 computers running for an hour, it's really 6 hours of work, but for you, standing outside the computer, it's really only 1 hour because they're all completing in an hour. So there's a little bit of a primer here if you want to expand it and read more about hail, but it's outside the scope of this discussion. But if you're interested, feel free. So the first step here is to grab our VCFs out of our workspace storage. So we're going to run this cell here. And what this is doing is pulling our VCF paths from our Google bucket. We essentially have a bucket filled with files, and we're turning it into just a list of paths so we can use that list later. You can see that the output is printing a couple of the list members so that you can see that it did work. This is a link to a VCF. The next bit is to import some more packages, and we're going to start a hail session. So run those 2 cells there. The first one is going to import some libraries that we need, and the second one is going to initialize a hail section. Now I will point out that this command will fail if you try to run it twice, because it says there's already a hail session running, which is fine, because if it's already running, you don't need to run it again. But just letting you know in case you try to rerun this workspace or this notebook again later without deleting your cluster. So that takes a minute, and then it has some output, again with that scary red background, but it's just telling you that it ran successfully and you've started hail. So, good job. All right, so once you have hail loaded up, our next step is going to be taking that list of VCF paths, and we're going to convert it into something that hail can easily access it in. In our case, it's a matrix table. For anybody who's not big into programming, there's a couple of different ways to reference a table in the traditional sense. So a matrix is a type of table. There's things called data frames that are a type of table. Broad strokes, it's like if you open an Excel spreadsheet and you had a bunch of columns and rows, but they have additional functions to them. So for this matrix table, it's composed of 3 parts. You have your sample annotations in columns, you have your variant annotations in rows, and then you have entries for your genotypes. And these matrices are optimized for fast access. So rather than storing a whole file in one place, it chops it up into little tiny pieces and stores it in a rapid access way. And that's going to allow it to access it quickly and work with it quickly. And again, it's part of that Spark parallelization there. So this command here is going to load when you run it. It's going to import the VCFs from this list of paths into a matrix table. It takes a little bit to run. And when it's done, we're going to run these next 2 cells. We've got mt.describe and mt.count. And I'm running both of them at once because mt.count is going to take a little bit. The reason it's taking a little bit is because we have 2,500 samples, so it's going to take a little bit to count them all up, I guess. In the describe section, we're going to see we have no global fields. We have your column field of S, which is a string, and I'll tell you now the S is for sample. It's giving you your sample IDs. Then you have your row fields, which consist of, again, we're looking at the genotype data. So our row fields are all about the locus of the variant call, the alleles, the RSID, the qual, the filters. You've got this info field, which you may remember from your VCF as having those, they're not filters. What do you filter on? Losing the word. Annotations. Yes. So you have all these annotations, and then for your entry fields, you have your genotype calls. So that is the structure of your matrix table. The next part is your mt.count. So you have your first number here and your second number you'll recognize as your 2504 samples from your data table. So you have 2,504 samples in your matrix. Then this 220,000 number. Anybody want to guess what that is? So if that's your number of samples, why would you have 220,000 of something? It might help to look at the describe portion. So if one count is the count of columns, which is your string, the other one's the count of your rows. So every row is going to be what? This is a hard one. It took me a while to figure it out. I'll just give it to you. It's going to be all of your variants. So each variant is going to have all this information about it in your call set since you're looking at VCFs. So you have 2,500-ish samples and 220,000 variants recorded in this. So our next step is going to be to merge our phenotype and our VCF data. So we have that table we loaded earlier using pandas, and we're going to translate it into HAIL. So using this table from pandas command in HAIL. Apparently they're used to doing this. So we're going to hit Shift Enter to run that one. And that's going to create a matrix table of samples. The next one is going to annotate the columns of our existing main matrix table, which is named MT. It's going to add this pheno portion to it, pulling from our new samples table we grabbed. So we're going to hit Shift and Enter to run that one. And then we're going to look at the first 5 rows of the matrix table. So this is querying the first 5 rows of HAIL. It takes a little bit to run. There we go. So looking at these rows, you'll see we have our locus, alleles, RSID. That looks a lot like the genotype information, right? From that portion. So, and as I scroll over, I get all the way to the end and I don't see anything labeled pheno, which we just added in. So where did that go? Well, I'm going to go here and I'm going to do insert cell below. You don't have to do this if you're not comfortable typing. I'm just going to go and do mt.describe. And what that's going to do is print out all of the fields that are in my table. So you'll notice that pheno actually ended up in the column field and not in the row. And it did that because we told it to. We told it to annotate calls or columns. Where is it? Yeah, there. Calls. So if we want to see the columns, all we have to do is, instead of printing out the rows, so I'm just going to copy this and create a new cell. Instead of printing out the rows, all I have to do is print out the calls, or columns. And it's going to take a teeny bit of time to run. All right, and now it's printed out the columns table of our matrix, which shows the pheno, age, ancestry, BMI, fasting glucose, all those things. And again, it's annotated by the sample ID. So matrix tables are a bit complicated to work with in this sense, but they are able to store the information in a very good way for data access. So now we have both our phenotype data and our genotype data in the same matrix. We're going to generate some variant level summary statistics using this command called variant QC. So it's going to compute some things like allele frequency, call rate, homozygous counts, and a bunch of others. So we're going to run this, and then we're going to take a look at how the table structure changes. So we're going to use mt.describe, and we're going to use this mt_rows show. So while that rows one is running, I'm going to talk a little bit about this describe section. Scrolling through, you'll see that the variant QC, since it's adding genotype level information, got added to the rows portion. So under rows, we have all the same old stuff we saw. We have this info field, but now we have this variant QC field. And it's a structure made up of a bunch of different calculations that were performed by our tool, variant QC. And it's all kept together in this nice little structure package that way. One thing I haven't yet mentioned is the fact that these annotations, so AC here, have this array int32 listed after it. That is the type of value it's storing in that location. So in this case, the AC, which I think is allele count, maybe no? No. All right. It's storing it in an array of ints, which is integers, so whole numbers. And if I were to query into that, I would see a list of various allele counts for that particular variant at that site. So it's very much like a table within a table within a table, but it's got really great ways to store information. Some other ones you have here are like just a full integer. I think there's some strings in here, which is just text, and then booleans, which are true and false values. But you can absolutely look through and if you're more interested in that. Now, down here in the shows command, if we scroll all the way over to the right, we'll start to see things starting with variant QC dot something, and those are all the items calculated with our variant QC command. We're only showing 5 variants at this point, but if you wanted to, you could explore more of the data set this way. Any questions on this so far? All right.
What does coerced almost sorted data set?
I'm sorry, which one? Coerced almost sorted. It's feedback that Hail is doing something. So that's like it reporting back, I've done this now. Now I'm moving on to my next step and I'm doing that. So when it's coercing the data set, it means it's wrangling it into a format that it can use to print this out. Yeah. All right, so then the last portion, I'd say, of this particular notebook of actual work we're doing is we're running 2 different steps. One is going to be a linkage disequilibrium pruning step, and that's going to pull out a set of variants that we can use for calculating relatedness of samples. And then following that, we're going to be using a principal component analysis to analyze the data and do that transformation in order to convert a set of correlated variables of genotypes between individuals into uncorrelated variables. And I'm sorry for reading my notes. I am not an expert in GWAS, so I am working on still understanding this, but ask me any questions you want, and either I can answer or we can get you the answer. So the very first step to this is doing some filtering. So first up, we're going to find out, we want to filter on the allele frequency, so we want to find out where allele frequency is stored. So we're going to hit mt.describe, and if we scroll down, you're going to find the allele frequency as one of the things that was calculated with variant QC. So this is the one that we want to target in our next step. Now down here, this command, which has already been written for you, is going to filter rows on our variantqc.af field, and it's showing index 1. So this particular AF field is an array, which means it has multiple things in it. I can tell you now that this array has 2 things in it. It has the allele frequency for reference sites and allele frequency for alternate sites. So you want the second one, which is our alternate sites, and for this case, that relates to index 1. It's a little confusing, but some programs use zero-based indexing, meaning the first position is 0 position, then you go 1, 2, 3, 4. So our second position is actually index 1. So we're going to filter that, and we're going to choose only the variants that have greater than 5% alternate allele frequency. So we're going to run that. And then after that, we're going to count the results. So we want to see how many variants remain in your data set after filtering. So before this runs, you remember from the first, we'd had 220,000 variants and 2,504 samples. So you should expect to still see the same number of samples, but probably fewer variants because we're only filtering out the variants. Just give it a little run. And that is what we see. So we have 2,504 samples still, but now we're down to 133,000 about variants. All right, our next step is to prune the variants using LD prune. So we want to, oh, go ahead. Okay. So this section is running an LD prune function. We're basically going to find a set of variants to run this on that are nearly independent of each other because we want to use that for our later PCA. So inputs to this one, we have an R² threshold, which we've set at 0.2. We have a window size, which we've set at 100,000, and then we have a block size of 1,024. So the block size at the end relates to parallelization, so you don't want to mess with that. But the other two, the R² and the window size, are basically how strict we are in our definition of independence. So if you want to, you can adjust those if you know what you're doing with it. We're going to leave it at its default settings for running this command. So when you're ready, go ahead and hit run on that one. It takes a little bit to go through, so we're going to sit on that for a sec. I've found that this command typically takes about 2 minutes, so if it takes longer than 5 minutes, that's usually because the kernel got frozen because I was talking too much in previous sections. And then you just need to refresh and run it again, and it'll run fast. So if ever you're working in a notebook and maybe you run a command that runs forever, there are commands like that, where you forget to put an end block on it or something and it just never returns. You have, in your terminal, you would do like Control-C interrupts, but in your kernel, all you do is go to your kernel menu and then say interrupt, and that's how you can stop whatever's currently running so that you can move on. Just a fun thing to know about. All right, so mine's returned. Has anybody else's finished? Yeah? We're still waiting on a few more? All right, give a few more minutes. If you're having any trouble, raise your hand. Let us know. If you have any questions, we can talk about them now too. All right, in the meantime, I'm going to run these next two commands to describe and count. After pruning, we've output the results to this variable called pruned variance. So when we describe it, we see a matrix table that's much smaller than our giant matrix table. And what it's doing is returning some locus and alleles for all the variants we want to prune down to. So when I go down to this prune variance.count, it says 25,409. So from our giant call set of originally 220,000 variants, we're only going to use those 25,000 for this next step. All right. Am I going too fast? Are people caught up? Good? Okay. Then our next step is to apply that filter of the prune variance to our matrix table. So we're going to run this step and then the count step to sanity check that it got down to here. We're doing that with this filter rows command. And basically we're looking in our prune variance table and asking, is the variant defined in our matrix table? And then we're filtering down to those ones. Yes? Okay, so we're using this filter rows command, and we're going to filter the rows down to whatever rows are defined between prune variance and they exist in the matrix tables the main matrix table we have. So if they're the same, we're going to keep them. If it's missing, then we throw it out. All right, and then after pruning, this count function outputs that we still have 2,500 samples, and now we have 25,000 variants. So that matches the pruning worked as we expected it to. All right, so now that we have filtered and prepared a matrix table, we're ready to run our principal component analysis, or PCA. So you run PCA using this function from Hale called HWE normalized PCA. And to it, you're going to pass in your matrix table, specifically your genotype field. And the K parameter is determining the number of PCs to return. So the larger your K, the more computation time this is going to take. So we're going to run this one. As a note, there are some other settings to it, like eigenvalues and loadings. Since we're mainly interested in the scores, we're only going to use that K parameter, which is our score return. So this takes a minute to run. Yes.
Like K
Because we want to return 5 PC values. This is what they chose for this analysis. If you choose more, you can get more PC values. If you choose less, you get less PC values. But that's what they chose for this one. I'm not fully sure unless, Laura, do you know?
We'll see it again in the semantic CMV tools also. If you choose more PCs, you can better model the data because each principal component that you add gives you more detail. But if you have noisy data, then those aren't going to be a lot of value, and you want to just kind of take the highest principal components because you can't model that detail anyway. So 5 is probably a happy medium, but for different data, you might want to try different things anyway. But this works well.
All right, so mine has finished running. Yeah, let's finish up this section and then we'll go to a coffee break. So we're going to run this describe command, and that's going to show us the PCs fields, and it's stored as a scores value, and it's an array. So our array is going to have 5 PC values, and we want to store them there. So in order to add it to our main matrix table, we're going to add it to our columns section, and we're going to grab the scores from our PCS table, PCS matrix, I should say. So that's what that step does. And then we, lastly, are going to visualize our samples. So when we run this step here, we're generating a scatter plot. It's going to plot our PC1 against our PC2 and divide it by the ancestry phenotype. So you get this nice little scatter plot here. So along the vertical is our PC2, and along the horizontal, way down there, is our PC1. And you can see different blob distributions of your different populations.
So yeah, if we usually visualize like the top 2, top 3, top 4. If you capture more principal components, you can sometimes find batch effects in your data. Like things, like if you're looking at exomes, then things that have different target captures might end up in different PCs. When we've done this for depth, for a different thing, we managed to isolate samples that did not have a common CNV. As you get into the smaller and smaller PCs, or I guess larger value PCs that make a smaller difference, it'll find more minute differences in your data, which is interesting. But adding more PCs isn't going to really change the results for this plot, for example. It's just kind of like, are you going to look at the rest of the ones or not? And if you're not going to, it probably doesn't make sense to do the compute to generate them.
Do you tend to do like an elbow graph to define how many components are you doing, or not necessarily for this type of data?
In practice, people will do that like the first time they do a particular type of analysis. Like if we, I don't know, if we started doing an association study on CNV data, which is something that we have the capability to do now, then we'd probably say, oh, how many PCs do I actually care about? Like how many batch effects are we going to capture? But like given that we've looked at genomes many, many times,
Right.
5 is good. Yeah, so the eigenvectors are the mappings of those PCs onto the original features. Yeah, yeah. How to actually get it from like code, that I don't know. I don't use Scale. But I'm sure it's possible. They have a really nice, are they near the docs? Is that what it is?
I'm not sure. If you expand the Hail primer, there's a link to their documentation.
Yeah, their main page is hail.is, but from there you can go to their documentation, which is pretty comprehensive.
All right, we're gonna take a quick coffee break. We're gonna come back after this to this same notebook. You can go ahead and pause it, and we'll just unpause when we get back. And this should save all the work we've been doing. It won't delete the files. So we're coming back at 3.
Chicos, un pequeño anuncio. Ahora durante el café van a hacer unas pruebas de alarmas. Pero no se asusten, es normal. Va a sonar muy duro, pero es que están haciendo deseando la prueba de alarma de incendios. Entonces, para que sepan que va a estar un poco ruidoso el café.
What's up?
An issue I'm having some trouble debugging.
Okay,
Kaden,
For whatever reason,
hold on.
there will be a simulation of the fire alarm. So don't be scared.
During this break?
During this break. So,
Okay, good timing for a break then.
yeah, it's because to avoid that noise during the course. So
Okay.
I prefer to make the simulation right now because there is the personal right now.
Okay. All right, let me come around.
Do you have intermediate files saved in that notebook that we could just jump in the middle?
yeah, so
What problem are you having?
what I'm seeing is the problem originated down here where we're actually pulling in our variant QC scores.
Okay.
If we look at when you do the describe of the empty object, there is no variant QC field.
After he's run the variant QC?
Yeah, I mean,
Where did he run the variant QC? It would be down, I think in section 6, maybe just above. Yeah, that one, 6.1.1. So you describe first. So this is where you're filtering, and it looks like it failed.
Yep.
Why did it fail?
Because there is no variant QC field in the empty object.
See?
Yep.
Oh, that's sensitive. Okay, so that's where you're filtering on it. Where are we running variant QC? It looks like you might have accidentally deleted something. All right, let me just look at what I have, because you don't have that command where you're running variant QC. So I think that's why it doesn't,
That would do it.
yeah. Okay, so right above empty.describe, you're supposed to have this line. So if you insert that, it should help.
What heading number is that?
I'm
just trying to navigate too much.
I don't know. It's like, it's right above where you apply the filters, so 6.1 area. I can't look at it without restarting the runtime, and it's going to take 2 minutes, so it'd be faster just to search. So it's this one, 5.2.4.
Oh. Yeah, that would do it. It's still. Yep.
Oh, there you go, it was collapsed. sound okay? All right, great. All right. As you're settling back in, please go up to the top right corner here and turn your runtime back on. You're just going to hit the play button. Looks like everyone's back, so I'm going to hit play as well. All right, let's get started here. So just as you were coming back, I asked you to restart your notebook runtime. If you haven't done so already, please click play. It'll take about 2 minutes to start back up. If you're still waiting on that, just follow along with me on the screen and you can catch up pretty easily. You should be on this screen probably is where it left you. We're going to hit edit again to open up the notebook. It's going to copy it to the runtime environment there. And we left off, I believe, in section 7. So if I go to the side here in our table of contents, I can click on that section just to jump down to it. So we finished up with this plot last time, and now we're going to move on to section 7. So we've done the analysis that we wanted to do in this notebook, and now we need to get it back out of the notebook into Tara's workspace that we're working in. So just as a quick refresher, actually, we've got this here. This is from the orientation slides back on day 1. And you have your workspace here. You have this data model where we pulled our phenotype data from that sample table, if you remember. And we have our workspace storage bucket. And this is where all of our VCF files and things live. But for our notebook, what we've done is we've created something separate over here, still in the workspace, this virtual machine environment of your notebook. And every task that you've run, every command that you've run that's produced an output file now lives in that notebook's storage directory. So since we want to continue on and run a workflow, we need the data not in a private notebook where it can't be accessed. We need it in our workspace storage. So this next section, we're going to go through converting that matrix table into something that we can put in our data model and put in our workspace storage. So back over to our notebook real quick. Up here. All right, so this first one is we're going to extract our sample metadata from the matrix table using the calls command. So that's just, did anybody else get that error? Yeah. This, all right. It looks like by pausing it, it deleted all the variables. So we're gonna have to rerun it. Can you think of another way?
I couldn't think of anything else.
All right.
You just have to go from the top and shift.
Nope, I got a better way.
All right.
Yes, so right here in this sample that we wanted to run here, we're gonna go up to the cell menu, and we're gonna say run all above. And what that's gonna do is rerun everything up to that point. It turns the whole table of contents red, which doesn't mean error. It means it's queued up to run. The yellow icon means that's the section we're selecting right now. But then as it finishes running these things, you'll notice the red goes away and it turns back to white. That means it's finished running. So this is probably going to take maybe a minute or two. And good to know that it deletes the variables. I thought it just did that for environment variables. All right, so it looks like it initialized a new Hail session. Okay, so it looks like it didn't actually delete the variables. It just shut off the Hail session, and when I restarted it, everything was already there. I'll have to explore that more later. This is the first time we run this particular section, so you guys are getting some brand new cutting edge content here. All right, this one is taking a bit longer than I'd like it to. So I'm just going to try interrupting the kernel and starting it again. So if you'd like to try that too, if it's also stuck at this pruned variants command, just go up to the kernel menu and interrupt it. and then do Shift Enter to run it. Yeah, the times it's gotten stuck for me before have been when I ran the whole notebook at once, so I think that's the issue with it. All right, there we go, now it ran. So for the rest of it, since we already did the run all above thing, we're just gonna need to do Shift Enter to the later commands. And you can actually Shift Enter through the text too, just to run it all. And then the last one you should be running is in section 7.1.1, this one. That's where we left off. All right, so mine has completed. Who's still waiting for theirs to complete? Okay, we got about half the room, so we're gonna get a little more time. All right. Show of hands, who's still waiting on theirs? Okay. Is it stuck at this step here, this prune variance? Yeah. Okay. Interrupt your kernel and try rerunning that command. I already tried. You already did that? Yeah. Okay. Then just wait. It takes like 2-3 minutes to run. Yeah.
Like I left it when we were breaking. I actually was in the hospital. Now I come back and it's still on black.
Yeah. You're going to go back to this step here, and you're going to shift enter all the way through section 7.1. Oh, okay. Yep. All right, so while yours is running, just follow along with me up here. So the next step we're doing is converting our data format. So essentially the table we just generated, the samples table, or matrix I should say, it needs a bit more post-processing in order to get it into a format where our data table in Terra will want to read it. So it's kind of outside the scope of this workshop to explain all that data transformation, but that's the code we're running for it. Essentially, here I'm going to hit Shift Enter to run it. Essentially, we're grabbing the S column, which was your samples, the phenotypes, and the scores from the PCA output. The next part is to flatten the PC array. So if you remember, scores is an array of 5 different scores. And instead of having it be like nested table arrays, we want it to all be at like the top level so we can see the scores easily. So what we're doing here is we're just creating 2 new columns to add to our samples table. We're going to grab the scores at the first index and the scores at the second index. And then we decided we don't care as much about the other ones in there. So we'll run this one. And once we've done that, we need to, this step is actually doing 2 things. First off, it's dropping the scores array. So since we've replaced it with its own 2 individual columns, we're dropping the array now because we don't need it. And then we're also converting it from a matrix format to a pandas format. So if you remember way back in the beginning, we created a pandas table for our phenotype data, and that one was much easier visually to look at and work with. So we're going to convert it to that. This is some additional converting that's happening. It's renaming the columns into things that we want the labels to be in our table. So we're doing that. And then this samples.head is just printing out the top few entries so that we can see that it looks like we want it to look. So in our case, we have all our phenotype data, which is the age, ancestry, BMI, all that stuff there. We have our sample ID as we expect. And then if I scroll over to the right here, you can see our 2 PCA scores that we chose to keep down at the end. So this is our table that we're going to bring to our data model now. And we're going to do that with this command here. So we're converting this table to a CSV file, which stands for comma separated value. And we're going to copy it from where it currently lives here in our workspace out to our bucket. So that operation should take very little time to complete. And the very last bit is we're going to add it to our workspace model. So if you remember initially from our exploration of the data tab, there was already a sample set there. So in order to do that easily, we've defined a function called makeSampleSet for you. So this isn't code you need to necessarily know too much about, but it's essentially creating a function for you to make this easily. And then this part right here is a little explanation of how to use it, but I like looking at this part right here. You're going to define a label, so this is what you want your sample set to be called. Now, the one already in there is called that, so let's rename it to something more relevant to you. So I'm just going to say GWAS analysis, and then my name. like that. It's going to create a phenotype file and put it in your bucket, so it's going to need that name there. It needs to identify a sample ID column, so that one can stay as it is. And then these last two bits are the portions that you may want to change, you may want to keep the same. This is saying this is the outcome we're interested in looking at, and these are the covariates we're looking at comparing it to. So if you wanted to design your own study using this data, you could examine a different outcome with different covariates. We had a whole bunch of options for you, and I think it's designed so that you can take this home and do some other options as well if you're interested in it. So other than changing the label, I'm leaving everything exactly the same, and I'm just going to run that. This one takes a little bit to do because it's creating something in our workspace.
Does the sampling technique that you're using matter?
Sampling technique?
Yeah.
I'm not sure I understand.
Like, let's say I don't know what kind of sampling you're using. Like, you probably like simple sampling, but if you were to do it proportionally to another variable, could you do it, or it's not really important?
I'm not sure. Laura, do you know?
When you take the sample, yes. Like a subset. Oh, okay. Yeah, because what I guess is just taking a random sample of rows with all the variables included. But if you were to, for instance, give a weight to the, I don't remember the name of the variable right now, but where the people is from, like Europe, Africa, Asia.
This part?
Like the ancestry, if you were to do it proportionally, for example, to that variable, could you do it, or it's not really important for the purpose of the analysis?
I think the LD pruning is what you're using to select a group that's, you're using it to select a group of variants that you can look at to determine relatedness of those variants, and that gives you those PC scores. So if you wanted to like proportionally select an amount from each population, you would do that instead of the LD pruning.
Okay.
I'm not super familiar with GWAS, so I think they might recommend using LD pruning over that option. But I couldn't tell you much more than that. Right. So now that we've created our sample set and this cell has finished running, our next step is to go back into the workspace, and we're actually going to run a workflow on this. So we've produced this output with these PC scores, and we want to use that for something. So there are a couple different ways to exit a notebook. You can hit this X button here. You can click on your workspace name. That's what I tend to do. Click on the workspace name. It takes me back to the dashboard. Once I'm in the dashboard here, I can go over to the Data tab, because I want to show you the stuff you just created. So you'll notice the sample set now says 2 instead of 1. And if I click on it, I can see my analysis is in there, as well as the initial one. So since I didn't change anything in terms of like the outcome and the covariates I'm tracking, it's got the same values, but it's creating stuff there. And now if you go over to Files, you see that there's this My Phenotypes CSV in there. And this is the table that you created and outputted. Now it lives here. So if I go over to the workflows here, you will see two different workflows. Now, for our sample set, we had a certain number of VCFs, and we've already run this step here for you. We had a group of VCFs from the 1,000 Genomes Project, and we ran that particular workflow to generate GDS files, which is going to be the input to our Genesis GWAS study. If you were to run this workspace, like copy it and put in your own data, you would need to run that particular workflow first so that you can get your GDS files, and then you can run this GWAS. But for our purposes, we can skip it since it already exists in our data model. So click on into the Genesis GWAS. And I believe this is the first time I'm showing you a workflow. Is that right? Okay. So there's a few different things to know about this page. Typically, all of your general information about the workflow lives up here. And then this is all down here, specific stuff that you can fill out. So up here it tells you the source, like where this came from. If you click on it, you're taken to a page where you can look at the script and things like that. But another way you can look at the script is actually right here in Terra. Click on the script tab, and you'll see this WDL script here that defines a task and then another task, and then I think at the bottom it's got a workflow. Yeah, they chose to put their workflow at the bottom here. Yeah. So this is the WDL code that this particular pipeline is running. So if you were interested, you could look through it and see exactly what commands it's running for every task and what it's doing. But for our purposes, we just need to know that we're doing a genome-wide association study. What we're going to do is process multiple workflows from a sample set. So it's already filled in with sample set, which means that's the type of value it's expecting. So if I go to this select data here, I'm going to select a sample set to run on. And since I went through the trouble of creating one, I want to use mine. But if you weren't following along or something errored, feel free to use this other one here so you can continue on with us. So we're going to hit OK. And now we have our data selected. It says one selected sample sets. This little checkbox here is for call caching. And call caching is a great little way to save money. Essentially, if I ran this workflow and it has 10 different steps to it, but maybe at step 5, something failed, like it wasn't working the way I wanted it to, and then all of a sudden the workflow failed and it stopped. I've already spent money running steps 1 through 4. I don't want to have to respend that if I don't have to. If I check this call caching box, it'll look in the history of workflows that I've run or that I have access to, and see if there are any other results out there that it can copy in. So in that case, when I run the workflow again after fixing the step 5, it would cache the results from steps 1 through 4, provided that nothing else has changed. I'm running the same samples. It's really intelligent about selecting when it can call cache and when it can't. It's going to just copy the results over, and in that way you're not spending the money to compute the results again and start over from step 5 where it failed. So that's a neat little way to save money. We're going to use call caching for it. Then the other 2 tabs down here are inputs and outputs. And I'm going to just take a quick look at this input section first. You're going to become more familiar with this on day 4, but essentially you have this workflow which tells you a bunch of different tasks, but then it takes in certain inputs. So in this case, we're taking in a GDS file, I think it was. And so we need to pass that file into here. And we do that through our pheno file. So this is our group of phenotypes, and it's pulling it from this dot, which means look in my table here that I've defined, which is our sample set, grab the phenotypes value. So just real quick, you don't have to go away from this page here. I'm just going to go over to my data tab here just to show you what that looks like. So it's going into this table and it's looking for the phenotypes, and it's going to enter in whatever you've put there. So if I wanted to, I could actually run on both these sample sets at once, and it'll run the workflow, and for one it'll grab this phenotypes file, and for the second one it'll grab this phenotypes file. And that's how you can run through and work with it. Wondering why this says 0 entities.
Yeah, I had that doubt. I thought I had done something wrong because the one that we
All right.
saves
Yeah, it doesn't have any
0.
samples. I'm going to explore why it does that, but in the meantime, just so yours runs the way we expect, we're going to grab this one, my SHG Terra analysis. So grab this one. I'm wondering if it's maybe because I do have to run that first workflow if we generated it ourselves. Might be that. I will find out. For now, go back to the select data and pick this one, not the one you generated. We're going to hit OK. All right, and then the other important part to take a look at is this output section. So this is where it's going to write all the outputs of your workflow. So workflows can have a number of different files created throughout the tasks, and as an output from that workflow, you can say, these are the outputs I care about writing back to my data model. These are the ones that I want to know about. And in that way, you can avoid having like 12 different steps of filtering VCFs when you're really only interested in the last filtered VCF, things like that. So the one we're most interested in is going to be this summary plots. So this .gwasp plots. That means our output is going to go to our sample set, which is our this, and it's going to go into the column GWASP plots. Now, if I'm looking at this, I don't see any column labeled GWASP plots. And that's because as an output, it can create its own new column. If I've run it before, then it would already exist there. And when I run it the second time, it would overwrite the value of whatever's in there. It doesn't delete the previous file. So you always have the opportunity to like go back if you need to, but it's just what's showing up in this table. So for our purposes, just leave everything as it is. It's all good. And we're going to hit this Run Analysis button. Now, it pops up and asks you to confirm just so you're sure you want to spend money on this, because this is going to launch a job to the cloud and it's going to cost us some money. For us, it's going to launch only one analysis because we're only launching on one sample set. It tells you what version of Cromwell you're using in case you wrote it for a particular version. It's a little bit of provenance there. And you're just going to hit Launch. Is everybody able to successfully launch their workflow? Seeing nods, great. After you launch your workflow, the first thing you're going to see is this page here. You'll notice it moved you over to the Job History tab. This is where you can go to both monitor currently running jobs and to check on past runs of jobs. It'll tell you that you have one submitted one, and the first thing it tells you is that it's queued. It doesn't automatically refresh, so if you hit refresh on your page, you can see if that queued status has changed. It might have, I'm not sure yet. Yeah, so now it's changed to submitted. So what that means is that Cromwell got your workflow, it packaged it up, and it sent it off to Google. So it's been submitted. And you can now click this View button here to go over to something called Job Manager. And you'll notice that opened up a new tab. So that one had an error. I'll have to report that to the Job Manager folks. They're interested in when that happens. Let's try closing that, and we're going to just reopen the view thing. All right, if you saw really quick here, it says it's in status running. Is anybody else getting this error, or is it just me?
No, I'm getting
It's asking you to log in? Okay, yeah, then I know what the problem is. I'm logged in on a different account.
Oh, okay.
So it's throwing an error saying I'm not allowed to access this different account.
So that means like for that group of samples,
So I'm just going to sign in with the other one.
No, the sample means like the individual, the person making the analysis.
Okay, now it works. Okay, so since it took me a minute, it's now running. And you can see that from the outset it tells you a list view of what tasks have been either executed or are currently running. So this task list will build as it continues on. You can check this inputs section to see exactly what it rendered your inputs as. So going back to the inputs configuration we were doing earlier, we saw it said like this.genotype files, and this is the value of this.genotypes value. File. So you can look and see what it rendered to and see if that was the correct thing you wanted. Labels is another thing that it gives you, telling you about the workspace and things like that. I don't tend to need those, but they're there in case you do. And then the next one over is a timing diagram, which currently is blank because nothing has finished running. So I'm going to show you one I ran earlier so that you can see what that looks like. Go over to job history, go to this one. We're going to view this. All right, you'll see that the list view now has 3 tasks in it. And then if I go over to my timing diagram, I see this really great chart of just multicolored bars. Now, I'm going to tell you right away, the colors don't mean anything specific. It just means that something different is happening in this yellow versus this purple. So the way you tell what it is actually by hovering over it. So if I hover over this big yellowy orange bar, it says user action. This is the part where your workflow task is actually running, it's actually doing something. These other things bordering it are about setting up. So localization means it's pulling files onto the machine. Container setup means it's starting up your Docker container. Pulling, analysis common, this is a whittle that it's pulling into the location. So various setup and then shut down things. What?
It's not a container.
Analysis common, Genesis whittle, that one. That might be pulling the container. I thought it was another whittle because it said whittle on it. I could be wrong.
Takes a long time.
Yeah, I can size
Could be.
Yeah, it's another opportunity to see if you're spending a lot of time localizing
Yes.
files, then maybe there's opportunities to use NIO instead of, like, to read the file out of the bucket instead of downloading it, because, I mean, that's time that you're paying for that the VM is
Yep.
buying.
Yeah, so looking at this, you'll notice that this task seemed to run on its own, and then these ones all ran at the same time, but after this one finished. So these ones were all running in parallel, but they all needed this one to finish before it did. So Cromwell is doing that intelligent thing where it decides what tasks can run in what order so that nobody has an error like, I can't find my input because it hasn't been generated yet. And then scrolling down, the very last task, it looks like it waited until the last one was done in order for it to run. So if I look here, it tells me that it took about 28 minutes to run this one. And this one is probably about the same amount of time it'll take yours to run, so check your results tomorrow because I'm not going to keep you here for an extra half hour to just see the results. This particular workflow was run with call caching not checked because I tend to have a lot of access because I've run these a lot of times before. So this is what it looks like when you're running it for the very first time. But when you've call cached something, let me see if my other job history one is a call cached one. Oh no, this one wasn't either. I was just running tests, it looked like. But if you run something and it's call cached, then it's going to have certain different things. So instead of saying user action, it'll say copying inputs and then updating call cache. So it takes a lot less time, which is nice. Yep.
The name of the tasks
Mhm.
are the ones that we use in
The Whittle script.
in
Yeah.
Okay.
Yep. So if I go back over here, back to my workflows, into the one we ran, and look at the script, you see one of the tasks is called Genesis Null Model, and this one has Genesis Null Model. So that's where it gets the name from. All right, and then it's nice to look at the timing diagram, but let's take a look at the output. So recalling that the output gets written back to our sample set, we're going to go scroll over and we're going to look at, where did it go? GWAS plots. So got this one, and this is what the plot looks like. Now, this is just showing you a preview of the file. You can download it if you want. It tells you a little bit about how much it costs, so less than a cent, which is good. But I'll tell you there's nothing more in this file than this image, so I'm not going to bother downloading it. So this is the output of our GWAS workflow. And you have your Manhattan plot down here, and I think that's an R curve. I'm not sure. I am not as familiar with GWAS, I apologize. But I do know that when you look down here and you see these dots, this is something significant happening above the level of baseline there. So these are the results, and this is what you would then take and interpret with your various studies. That's all I have for you today. Are there any questions about this? Or any questions about germline topics in general? This is the last germline day we have. Tomorrow we're going to start up on somatic. You can still feel free to ask us germline questions tomorrow, but.
Tomorrow we're
Yes.
gonna get these plots for this running, right?
Yes, you should have the plots done by tomorrow. And then just to go look at them, because we're not going to review them all together, you'll just go into this workspace, go into your data tab under sample set, and you'll be able to see it there. Yeah?
But this case, the probability of more variance in one section of the chromosome?
Not sure. I know that we generate this plot, but I don't know how to interpret it yet.
For Manhattan?
Yeah.
Yeah, you get your, I remember, goes back in the day. Because I know that canine genetics people used to do them a lot. So you had a Manhattan plot, and you're supposed to have your baseline snips, and then you have your sites that are the most of interest. So a good Manhattan plot looks like the sky, it's not like a Manhattan, so you have like high places. Yeah, you shouldn't look like Durham. So you shouldn't have like tiny mid-sized buildings. That is actually a running joke. If you have a Durham plot, then your GWAS sucks. But if you have actually a Manhattan plot with tall places, then you're doing it right. And the taller they are, and that's actually then you have that probability on your y-axis. So those are the best candidates that you have. Something in that line. I haven't worked or done anything GWAS associated with that. And then on your x-axis, you have chromosomes, the different chromosomes. Yeah. So in humans, you would have your human chromosomes, and then in dogs, you would actually have 24 because they have 4 chromosomes. I'm a little rusty in that, but yes.
The probability of variance or sleep?
No, it's the association with that trait of interest. Because
So
if I stand so you have a disease, Addison's disease. This is real because I still remember that one. With Addison's, actually, you had it was a Durham plot actually, because you had so many very low, very close to the threshold line, so you really didn't have any potential sites that were associated with Addison's until they basically had to expand their cohort until they found a potential candidate that actually was high up there. So that's kind of in theory what you were looking for, something that is up you have your threshold line, then you have something that's way out there, and that would be like your best case scenario. Because genome-wide associations, traits is SNP associated or a set of SNP associated with
So in this case then, since we were looking at BMI, these would be candidate sites of interest. This one would be candidate sites of interest for variants that may affect BMI. Is that what you're saying?
I'm sorry, I just
No, it's fine. So those 1, 2, 3, 4, 5 points that go way up, those would be candidate variant sites for influence on that BMI trait that we were tracking.
That would, if that's,
Yeah.
if that's
That was
the
the thing we
variant
were-
of interest that
yeah.
you're looking at, yes, if that's your response variable that you're looking
Oh, okay.
So in your experiment design, then you would actually have your response factor, which would be that your BMI, that would be the case.
Yeah.
Then yes, you're looking at that. Then it would be those, like if that's your genome-wide SNPs, then that would kind of be like that. Yes.
Okay.
They kind of did the same thing with Addison's. So you would actually do that. Oh, there are other, I know that there's many more things that you have to tweak around that. I'm the same boat.